that this epithelial lining is only derived from the endoderm. That is, we are saying the GIT develops from the endoderm. So be very specific that you should always remember that this epithelial lining is, for in this case for the GIT, the epithelial lining is only derived from the endoderm. Okay. The other layers, these are not derived from the endoderm, they can be derived mainly by the mesoderm. Okay, like this submucosa and then muscle layers. Of course, you can understand muscles means they will be derived from the mesoderm, and in there we have the blood vessels present, the lymphatics we have. So these are basically derived from the mesoderm, and over here there are certain nerve plexuses. So these nerve plexuses, you can understand they are part of the ectoderm. Okay, now always remember that this mucosa, whenever you're talking about the mucosa, remember that mucosa consists of three layers. One is the innermost layer is the epithelial lining. In this case, this is derived from the endoderm. Then we have a layer that is the lamina propria, and there is the muscularis mucosae. So we are getting over here, actually we are getting three muscles. When we reach the stomach, remember stomach as an exception, stomach also has an additional layer that is in the part of the muscularis externa. So these two layers, if you talk about these two layers are basically coming under muscularis externa. Okay, and the internal muscular layer, that is a part of the mucosa, remember that is muscularis mucosae. Okay, so this difference should be very clear. This is very much required for your embryology and anatomy part also. So this should be kept in mind that mucosa itself is divided into three types, means it has actually three parts. One is the epithelial lining, which is the innermost layer that actually circles or covers the lumen. Then we have a middle layer that is the lamina propria, and we have the muscularis mucosae. Now first let's talk about in very short, let's discuss about the origin and development of the GIT. Okay, now over here we are coming to the development of the gut tube. So remember, if you have read embryology, then you should be familiar with what I'm talking about, that whenever there are mainly two types of foldings that we are getting over here. One is the cephalocaudal folding, okay, and one is the lateral folding. So due to result of these two foldings, okay, one is the cephalocaudal folding and one is the lateral folding, the embryo which was actually formed, if you remember, there was this embryo was formed and embryo was actually folding itself. So whenever this endoderm, already there is differentiation into the three layers, already ectoderm, endoderm and mesoderm, that has been formed. So after that, when we are coming on to this part, remember that the endoderm is mainly forming the primitive gut tube. Okay, the primitive gut is from the endoderm, and mainly now we are concerned with the epithelial lining which is coming from the endoderm. The yolk sac and the allantois, they will remain outside. So remember that during the gastrulation phase, endoderm during gastrulation phase, the phase is actually gastrulation. So during this time, this is forming the primitive gut. Okay, this is forming the primitive gut, and at that point of time, the yolk sac and the allantois, they remain outside the embryo. Now in the cephalic, one is the cephalic part. You can understand that if this is there is a head, there is a tail. So the head part is actually called as the cephalic part. Okay, and the tail part is called as the caudal part. So as I've told you, there is a cephalocaudal folding. That is, folding is happening like this way. Okay, so whenever this will be there, a certain portion of the endoderm will get incorporated within the embryo. Might be that here I'm not discussing in details, but if you have attended your embryological classes, then you will be familiar with what I'm saying over here. So remember, during this type of folding, the yolk sac and the allantois that will be formed, they will remain outside the embryo. In the cephalic and caudal parts of the embryo, the primitive gut actually forms a blind ending tube. So there is formation of the foregut and the hindgut. So whenever they will ask you about the what is this gut and the different forms of the gut, so remember gut, this is mainly three types. One is basically the foregut, one is the midgut, and one is the hindgut. Now in your exams, they will ask you, or in the viva table, they will ask you about what is the extension and all that. I will be discussing a little portion of it. So remember that when this was formed, mainly there were two blind endings of this tube. And in this middle part, this midgut part, this remained temporarily attached to the yolk sac. There was basically this, suppose this is a yolk sac, and there was temporary attachment of this midgut, okay, with the yolk sac by a specific duct that is called as the yolk stalk, or you can also name it as your vitelline duct. Okay, this was the vitelline duct. So remember, this is temporary. Okay, here I'm not writing properly, but remember this is temporary, and there was this attachment of the midgut with the yolk sac. Now the primitive gut tubes and derivatives, they have four sections that you will be knowing. I'm not going into these details. There will be the pharyngeal gut, which is basically extending from the oropharyngeal membrane to the respiratory diverticulum, and that becomes a part of the foregut. And the remainder portion of the foregut will lie caudal to the pharyngeal tube. Then we have the midgut, which begins caudal to the liver bud and extends to the junction of the right 2/3 and left 1/3 of the transverse colon in adult case. So this is the extension of the gut tube that is generally asked in your embryology. Then there will be the extension of the hindgut, that will be from the left third of the transverse colon to the cloacal membrane. Remember, this endoderm, this mainly forms the epithelial lining, as I have told you, and there it gives rise to specific cells, the parenchyma. Okay, if you are talking about this endoderm, then this endoderm, from this endoderm there will be origin of the epithelial lining. Okay, the epithelial lining will be formed from here, and remember that this gives rise to the specific cells, that is the parenchyma. So this forms the certain cells, they are the parenchyma cells. Now they are having different differentiation, they will differentiate, like there can be the hepatocytes from this. Okay, then there can be the endocrine exocrine cells of the pancreas, and the stroma, the connective tissue part, that is derived from the visceral mesoderm. Remember this, I'm writing this point, that whenever you are talking about the stroma, this stroma is basically a part of the connective tissue. Okay, and this, always remember this fact, that this is for the glands. Okay, like it can be the liver, it can be the pancreas. So the stroma is derived from visceral mesoderm. So this can be asked as an MCQ question or sometimes fill in the blanks, or in the Viva table the teacher may ask you directly. So there is the visceral mesoderm, and remaining there the muscles, then there is the connective tissue, then there can be the peritoneal components of the wall of the gut. They are derived from the visceral mesoderm. Remember this fact. Now coming on to the mesentery. Here, if you have seen this picture carefully, then here you will find that I have written something, that is, what is this mesentery? Now let me discuss over here that this mesentery is basically a double layer of peritoneum. Okay, double layer of peritoneum. So certain organs will be actually, when there is folding, so this folding will result in this mesentery. What will happen? Certain organs that will be developed during gastrulation or embryogenesis. After embryogenesis we have organogenesis. So in the organogenesis phase, remember, when organs are formed, then this peritoneum in the gut tube, when this gut tube is formed, certain organs will remain suspended inside. Okay, they will be intraperitoneal. So this mesentery actually gives covering. When there is this double layer of peritoneal folding covers the organs, then they become intraperitoneal organs, and certain structures remain outside. Okay, they are the extraperitoneal structure. One fine example is a portion of the kidney that is basically remaining extraperitoneal. Now this mesentery, which has a collection of connective tissue that is maintaining the gut tube, because through this mesentery the blood vessels, the lymphatics, the nerve supplies, they are actually getting connected to the gut tube. Okay, so the gut tube is basically, this mesentery is acting as a stalk, or it's a connection medium. Okay, that is connecting two structures. One is this gut tube and to the body wall. Okay, from the body wall, if you have learnt about it in the embryology classes, that how this thing is been formed, then you will understand that there is a body wall at the back over here, and the body wall is connected with this gut tube via the mesentery. Okay, the mesentery is the stalk or connection, and through this there is passage of the blood vessels, there is passage of the nerves, there is passage of the lymphatics and other structures. This has also two parts. One is the ventral mesentery, one is the dorsal mesentery. So the dorsal mesentery will extend from the lower portion of the esophagus to the rectum as a continuous sheath attached to the posterior body wall, and this will provide the passage to blood vessels, lymphatics and nerves, as I have told you. Then these regions included in the dorsal mesogastrium, remember that the structures that these regions will be included in the mesentery portion, that is in the dorsal mesentery. If I talk about, then remember this mesentery is divided into two parts. Mainly we have the dorsal mesentery and one is the ventral mesentery. Now in this dorsal mesentery, remember that certain regions are included in this. They are the first, remember, this is the dorsal mesogastrium. Then we have the greater omentum. What is omentum? This you will know later when we will be talking about stomach. Then we have mesoduodenum, mesocolon, mesoappendix, like this. Okay. So I'm not going into further details, but remember that this mesentery is very important structure connecting them. If you are talking about ventral mesentery, remember its origin. It is derived from the mesenchyme of septum transversum. Okay, so this ventral mesentery is derived from mesenchyme of septum transversum. And growth of the liver into the septum will actually further divide it into two parts. So liver bud actually grows over here, and this will be further divided into two subtypes. First is the ventral mesogastrium, and that is also called as the lesser omentum. So these omentum, these are basically folds of peritoneum covering the stomach. Okay, so here we have got the dorsal greater omentum, which is a part of the dorsal mesentery, and we have got a ventral, from the ventral mesentery we have got the ventral mesogastrium or the lesser omentum. Okay, which will be extending from the stomach to the proximal part of the duodenum and also to the liver. And another part is basically the falciform ligament. Now might be you are familiar with this term, falciform ligament. This is basically a ligament that is passing between the two lobes of the liver. Okay, we have mainly, if we see, this ligament actually divides the liver into two halves. One is the right half and the left half, mainly the right lobe and the left lobe. Okay, so that is the falciform ligament, which is extending from the liver to the ventral body wall. So remember this much is enough. Now I'm not going into the boundaries. I have just told you, if you can recall, I have told you about the extensions of the foregut, the midgut and the hindgut, which can be asked as your Viva question. Now moving on to the physiology part, let's discuss about certain muscles. Okay. Now when we're talking about the types of muscles, remember there are mainly the smooth muscles present over here. So smooth muscles, they can either act as a single unit smooth muscle or they can be a multiple unit smooth muscles. So these are basically, what is this multi-unit smooth muscle? This is basically, it is having discrete separate smooth muscle fibers, and each of these fiber is actually independent. Okay, they are innervated by a single nerve ending, so each fiber actually becomes independently operating, and they are actually innervated by a single nerve ending. So remember this thing, that on the outer surface of these fibers, there will be certain fibers which will be like those of the skeletal muscles, and there will be a thin line layer of basement membrane with a mixture of fine collagen and glycoprotein, okay, on them. And then we have another type of smooth muscle, that is the unitary or the single unit smooth muscle, which is basically, that is a syncytial smooth muscle, so we call it the visceral smooth muscle. So what do you mean by syncytium? Syncytium means that is a cell which is having multiple nucleus. Okay, remember this, that whenever we are saying that there is a multinucleated cell, that is basically called as a syncytium. And there are individual, like there can be 100 to 1000 muscle fibers which will be acting as a single unit. That's why it is called as a unitary or single unit fiber. And these fibers will be joined by gap junctions, so that the ions can flow freely from the muscle cell to the part, and there can be also action potential generation. Okay, Now, gastrointestinal smooth muscles, they function as a syncytium. Smooth muscles of the GIT, they will function as a syncytium. Each fiber, which has an approximate length of 200 to 500 micrometer and 2 to 10 micrometer in diameter, so they are arranged in bundles basically. Okay. Now, as I was discussing, that the muscularis externa, that has mainly two muscle layers. In the stomach, we have an additional muscle layer, that is the oblique muscle layer. So over here in the GIT, we are seeing that muscles are arranged in this fashion. So these muscles, which are arranged mainly, if you see, this is the tube is placed like vertically. Okay, this tube is placed vertically. So if you see these muscles, these are arranged what? These are encircling this tube as if. So they are basically the circular muscles. And we have longitudinal muscles also, which are arranged in this vertical patterns. And mainly when we are taking the cross section, we see that how these muscles are arranged. The outer layers, they are longitudinal fibers. So whenever there will be longitudinal fibers, you can understand that whenever longitudinal fibers you will be seeing, so you will be seeing like this type of dot 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 structures. And whenever you will be seeing circular type, so in the circular type you can see like this type of, okay, like this type of fibers are encircling. Okay, whenever you take a cross section, if you divide this GIT like this, if you take the cross section, then you have seen the diagram was in the first, this diagram which I was showing. So it comes out almost like this, and over here the longitudinal fibers, they appeared like this dot 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 fibers because they were longitudinally placed, they were vertically placed, right? And these circular fibers, they appear like this. So this you should keep in mind. The function of gap junctions present in this case for the GIT smooth muscles, as they are basically they are syncytial or multinucleated smooth muscle. This is very important because action potential generates at one point and travels in all directions. So that's why remember that the smooth muscles of the GIT, they are syncytial smooth muscles, or they are mainly the unitary smooth muscles. What is the function of gap junctions? Remember that there is low resistance to the movement of ions from one muscle fiber to another. So there is a smoother conduction of the action potential. Now coming on to the electrical activity, remember that in the electrical activity there are mainly two basic types of electrical waves obtained. One is the slow wave and one is the spikes. So these slow waves, whenever, remember that the GI contractions, they are basically rhythmic, and whenever they are happening in this particular rhythm, they are actually determined by the frequency of slow waves. So these waves are actually not the action potentials, but they are the slight undulating changes in the resting membrane potential. So whenever a cell is at rest, it attains a certain potential that is called as the resting membrane potential. So in this case, for the muscles of GIT, they have a resting membrane potential of 5 to 15 millivolt. Okay, so there is actually, these waves are generated due to certain contractions, which are rhythmic contractions, and when there is rhythmic contraction, there will be certain frequency of slow waves obtained. May be caused by specialized cells. There are certain specialized cells, like we have the interstitial cells of Kagel, which are present, and they are basically the electrical pacemakers for the smooth muscle cells. Now talking about, in this case, let's talk about the spikes. So these are the two action potentials which are automatically occurring when the resting membrane potential is becoming more positive. You know that the resting membrane potential for a now muscle cell, which was like about -70. So from here, what will happen? From -70 millivolt, this will actually seem to rise. It will cross 0 and it will come on to approximately 35. Okay, it can also range to 40, 45. But mainly remember that from this, what will happen inside the cell, there was this negativity is obtained, and whenever there will be positivity inside the cell, that is, there will be an action potential generated. So the potential inside the cell, that will rise from -70 to +35 like this. So in this case, that was for a nerve muscle. This I had this is for a nerve muscle. Okay, so in this case, we are talking about the smooth muscles of the GIT. So they have a range of -50 to -60 millivolt. That is the resting membrane potential that was present. Now whenever this two action potential comes, there will be the higher value of this wave potential, and there will be also increase in the frequency of the spike potential, and that is directly proportional. That will be approximately about 1 to 10 spikes per second. This effects, as I have discussed, that this slow waves effect is mainly seen in the muscles of the stomach. Okay, stomachs, as I told you, they have an additional muscle, that is the oblique muscle. Now factors depolarizing this membrane. So whenever there will be stretching of the muscles, whenever the muscle will be stretched, at that point of time there will be an action potential origin. Then ACH stimulation, acetylcholine, which is basically released from the parasympathetic nerve endings. So ACH will actually stimulate, and that will actually cause action potential, and there will be depolarization of the membrane. And there will be also stimulation by certain GI hormones. This I will discuss later. What are these GI hormones, the names, example, their functions and all. So remember acetylcholine and noradrenaline. Okay, these have mainly certain effects on the smooth muscles. Talking about the neural control of GI functions and the enteric nervous system, which is, remember that more than 100 million neurons are present in the walls of the GIT. This is a fact, that more than 100 million neurons are present in the walls of the GIT. Now additionally, we have a CNS, we have peripheral nervous system, we have central nervous system. Under peripheral nervous system, we further have like the somatic, the autonomic. Autonomic is further divided into sympathetic, parasympathetic, that is there. Okay, but to maintain and control the GI functions, we have a separate nervous system that is called as the enteric nervous system. Okay, now enteric nervous system has a plexus. Two types of plexuses are there. One is the myenteric plexus and one is the Meissner's plexus. Myenteric or Auerbach's plexus, one type, and Meissner's or submucosal plexus is another type. So we have these two types of enteric plexuses present. Myenteric plexus is located between the longitudinal and the circularis muscles of the muscularis externa, and Meissner's plexus, by name, it is understandable that they lie in the submucosa. So in the submucosa, there are certain submucosal glands like the Brunner's gland. Then certain other glands are present which have their respective functions. So remember that any stimulation sent by this Meissner's plexus, they will be affecting the secretions of the glands, submucosal glands. And whenever there is any type of words about the tone of the GIT, okay, these are related to the muscularis externa mainly. That means longitudinal circular muscles. That means you will find the function of the myenteric or the Auerbach's plexus. Remember this. Remember, whenever there is a sensory stimulus originate from the GI epithelium, then afferent fibers will actually send them this stimuli will actually be sent to both the plexuses, that is the Auerbach's plexus and the submucosal plexus. Then there we have the prevertebral ganglia of the sympathetic nervous system. There also afferents will be sent. Afferents will be sent to the spinal cord, and also via the vagus nerve they will be sent to the brainstem. Okay, now let's look at the differences between the meningeal and the submucosal plexus. Let's see. So over here, as I've told you, meningeal plexus, where will it be found? It will be found between the longitudinal and the circularis muscle. And submucosal plexus, this will be found in the submucosa itself, that is the Mesenous plexus. So here you remember that under meningeal plexus it is mainly controlling the muscle activity along the length of the GIT. So that means anything related to the tone of the gut wall, then intensity of the rhythmic contractions that are happening, or there will be certain increased velocity and conduction of the excitatory waves along the GIT. So these will be all related to the meningeal plexus, whereas in the submucosal plexus that will be in the inner segment, that is towards the submucosa, in the submucosa. So that will be related to the intestinal secretion, absorption or contraction of the submucosal muscles, all this. And mainly submucosa has excitatory neurons. Remember this point, that mainly submucosal plexus has excitatory neurons, whereas the meningeal plexus has both inhibitory as well as excitatory neurons. Okay, and there will be certain inhibition. There will be, of course, the intestinal sphincters will be there, the closing of the pyloric sphincter. So these will be all under the control of the meningeal plexus. That is very important. So this diagram, look at this diagram, how these fibers are arranged. Okay, it is not asked in details, but sometimes this difference can be asked in your viva table. So remember this point. Types of neurotransmitters secreted. So if you ask about any type of neurotransmitter name, if they're asking simply in the viva table, if the teacher is asking you that give the names of the neurotransmitters secreted by the enteric neurons, freely you will name these: acetylcholine, norepinephrine, even ATP, serotonin, dopamine, somatostatin. Then we have others also, like we have the bombacin, the neuropeptide Y. Here I'm writing, okay, bombacin. Then we have the neuropeptide Y. So these are certain other types of neurotransmitters secreted. The nitric oxide. Now the autonomic control of the GIT. So autonomic control, this is an additional control that is coming from the sympathetic and the parasympathetic. So parasympathetic, that is, parasympathetic has a cranial sacral outflow. So from there this is control, and under the sympathetic, that is basically the thoracolumbar. So from T5 to L2, this is coming the sympathetic plexus, that is mainly originating from the lateral horn, mainly between the thoracic and the lumbar segments of the spinal cord. So there is the lateral horn. From there the fibers are going to the sympathetic chain, that is, this is the sympathetic column of the sympathetic chain, which is running parallel, almost parallel to the vertebral column, which has basically sympathetic chain that is relaying further into two ganglia. One is the cilia ganglia and one is the mesenteric ganglia. What are the afferent supply? So that was the autonomic supply. Now talking about the afferent supply, that is the afferent sensory nerve fibers from the GIT. They have the certain cell bodies that are located either in the enteric system or they are located in the dorsal root ganglia of the spinal cord. Sensory nerves, they are mainly stimulated if there is any sort of irritation. Now if you are constantly, if there is irritation, like in cases of GERD, gastroenteric reflux disease, so in this case what will happen, there will be reflux of the acid mainly into the lower portion of the esophagus. So in cases of GERD, what will happen, there will be change in the cell lining of the lower end of the esophagus. There will be changes, and there will of course be irritation. So these irritation impulses will actually be sensed by the sensory nerves present. And in cases of excessive gut distension, that can happen if you have eaten extra amount of food, so there will be excessive gut distension. A very short topic might be asked in your viva table, that is the gut-brain axis. What do you mean by that? So gut-brain axis, so that is talking about GIT and brain, and there is axis means that is talking about a connection. So it's a link between the CNS, the ANS, enteric nervous system. Then we have another thing, that is the hypothalamic pituitary adrenal axis, HPA. I'm not going into details, okay, because this is not very frequently asked in your examination. What is the function of the gut-brain axis? Mainly to maintain and monitor, because of course there should be an intervention. When we have a separate enteric system, it's good, but there is an intervention of the CNS. So there is to maintain and monitor the gut functions and integrate other functions with the body. The GIT secretory glands are there, certain secretory glands. One is for the digestive enzyme production, and one is the mucus glands, which actually produce mucus. Remember the spelling, mind the spelling, okay. Mucus, this is basically talking about the secretion, and when you are talking about the gland, then it will be, you write at this O. Digestive enzymes, we have the salivary gland, we have the liver, then we have the pancreas. So these are the other glands, digestive glands, which are mainly producing the digestive enzymes. Next we will discuss later about the salivary glands and secretion. So before that, just I'm showing you the slide over here, that what we have discussed till now. This electrical activity, there are other things I'm not going to details which are not asked, like the conduction and how action potential, that is stimulating the smooth muscles, that is generally not asked in your papers. So I'm not going into those details. I'm just talking about in GIT what is happening, that the smooth muscles of the GIT, they are the syncytial muscles, okay, where when action potential will be generated at one point, it will spread and travel to all directions and all. I have discussed this diagram, which will not be asked in your exam, but you should know that what are the layerings of the GIT. And little I have discussed about this thing, that is the mesentery, how GIT was developing, okay. That is from gut. This difference is asked mainly in the embryology paper, foregut, midgut, hindgut, their extensions. And remember this point, that endoderm is forming only the epithelial lining of the GIT and not other structures, okay. And in the epithelial lining there will be hepatocytes and other structures associated, and the remaining stroma portion or the connective tissue portion is derived from the visceral mesoderm. So this was about our introduction to GIT, and we have discussed certain other points also regarding the origin and development of the GIT. Thank you. Hello everyone, I am Saptarshi Viswas from Murshidabad Medical College. Last day we had discussed about the introduction to the GIT, we had discussed about the layers of the GIT, we had talked about the valves, about the muscles of the GIT, and also a part of the development of the GIT. Now last I was discussing that there are certain glands present in the submucous. They are the submucosal glands. So the root of the GIT, that is starting from the mouth to the anus, in this entire tube there are the submucous glands present at certain places where the secretions are added. But there are also associated glands which are responsible for digestion and absorption to some extent. So that happens because of these glands. Like there are the salivary glands, there is the liver, and also we have the pancreas. So these glands are associated glands with the GIT, and the secretions are very important for digestion and absorption and that entire processing of food. So remember that whenever we are talking about the enzymes of liver, the enzymes of these salivary glands, you should be very specific about it because they are having a specific function. Now coming on to the salivary gland and its secretion, whenever the food is coming in contact with the gut epithelium, that is whenever you are taking the food in your mouth, so you are actually chewing the food, there is mastication happening, and whenever this mastication will happen, or there is the smell of the food, at the smell of the food also you will see that you will actually start salivating. If you that dish is known to you, you know the taste of the dish already, then you will start salivating. You know that this particular taste is of that particular food and you have already tasted it, so your brain has that information, it has that memory, and now it will start your salivation. Okay, you will start salivating. So whenever the food is in contact with the gut epithelium, the enteric nervous system gets activated and there is release of saliva from the glands. Okay, so that sensation could be a tactile sensation, there can be chemicals irritation because these food substances are chemicals, or there can be distension of the gut wall. Okay, so distension of the gut wall, that will also happen when you have taken extra amount of food, so automatically the enteric nervous system will get activated. So that is very important. Now coming on to the parasympathetic stimulation, remember the salivation is mainly under the control of the parasympathetic stimulation that will increase the glandular secretion. So this saliva, which is salivary glands, they will start producing saliva. Coming on to the sympathetic stimulation, now remember sympathetic stimulation has dual effect. Dual effect means that at times it will slightly increase. Okay, in sympathetic stimulation there can be slight increase in saliva production, or if there is already stimulation by the parasympathetic or due to any hormone, then what will be the function of the sympathetic system? It will try to reduce it. Okay, so it has a dual effect that you should remember. The parasympathetic system is mainly increasing the glandular secretion, whereas sympathetic system is having a dual effect where it can slightly increase at times, or when already parasympathetic or hormonal system they are already stimulating the secretion, then what will the sympathetic system do? It will try to reduce the secretion. Now see, this is very important mechanism of secretion by the glandular cells. Now see what is actually happening. The nutrient material required for formation and secretion first diffuses by active transport. From where will this diffusion take place? This diffusion will take place from the blood. Okay, from the blood there will be diffusion. So these active nutrients, so I can write over here that nutrient material for the formation of secretory material, that is for the secretion, that from where will it be coming? This will first diffusion from capillary into the glandular cell. Nutrient required for formation of the secretion will diffuse from the capillary to glandular cell. So this should be very clear. Next, we are seeing that there are certain mitochondria present. Okay, these mitochondria you can spot over here. These mitochondria are present. So what is the function of mitochondria? We'll all know that it is mainly. So what will the mitochondria do? They will produce ATP because energy production is very important for the cellular functions. Then what will happen? They will actually secrete these organic substances mainly via this nuclear. In the nuclear material there will of course be certain formation of proteins. You all know there will be transcription, translation, and ultimately proteins will be formed. So they will actually get they will actually come to this endoplasmic reticulum, and this endoplasmic reticulum has also ribosomes attached to it. So ribosomes will act as protein factory. So there will be secretion and also certain packaging and modification. They will all be done by the Golgi apparatus. So in this Golgi apparatus which are present, so here the modification and packaging of all those products will be done, and they will be stored in this zymogen granules. These are basically what? These are actually secretory vesicles. Okay, these are the secretory vesicles, and what will happen that this particular substance, the secretion, will actually be stored in the secretory vesicles, and they will be released from time to time. That will be released from time to time. Okay, so this process should be very clear that these vesicles actually are stored for the nervous or hormonal control signals that will be coming. So how this will happen? The hormone will bind to the receptor. Now let me write this part. So hormone will actually bind to the receptor that is present, and what will happen? It will express itself actually. So this will possible, there will be the increase in the conduction of this hormone into the cell, and the machinery will go on, which will actually cause increase in cell membrane permeability. There will be increase in the cell membrane permeability to calcium ions. Okay, so calcium ions will actually become more permeable. So calcium will enter into the cell and fuse with the vesicles. So what will happen? Calcium fuse with the vesicles, it will enter and it will fuse with the vesicles. This zymogen, what do you mean by zymogen? Zymogen is actually inactivated enzymes. So these are stored in the secretory vesicles. So their calcium will fuse with these vesicles, and what will happen? They will fuse them to the apical membrane. Vesicles fuse to apical side. Okay, this is the apical side, this portion is the apical side. So here the vesicles will fuse, and what will happen, there will be breaking or rupturing of the membrane. Okay, and there will be exocytosis. So this is very important mechanism. Whenever you're asked about the glandular, this is just for your idea that whenever there is glandular secretion, this is happening in this way. Nutrients are first diffused from the capillary side into the cell, and then the mitochondria will produce energy. Then there will be certain translation all will happen in this nucleus, and from there the products will actually go to the ribosomes. There will be production of proteins, and also attached with them is the Golgi complex, where there will be further packaging and modification, and then they will be stored in the zymogen granules, and from there this hormonal control signal will come. They will cause increased permeability to calcium. Calcium will fuse with the vesicles. Vesicles will then fuse with the apical membrane, and ultimately there will be exocytosis. So this should be very clear. Now remember, mucus is produced. Mucus is very important. Mucus is always produced by the saliva, and the secretion of saliva, most part of it is water. We all know there will be electrolytes and several other glycoproteins present. So this property is mucus. What is this property of the mucus? Let's discuss. So mucus has adherent qualities. Okay, that will mucus. What will it do? It will adhere to the food particles. Mucus will adhere to the food particles, and it is sufficient. The body will actually, it will coat the food particles. So adherence to the food particles, then coating of food particles. Food, so here it actually passes as the food bolus. So bolus will be coated, and what will it do? It will actually make it slippery. That means it will cause, mucus has low resistance. So mucus, it will make food bolus slippery for easy movement down the esophagus during deglutition. So when you will swallow the food, at that point of time the mucus will actually make it coat the food bolus. It will adhere to the food particles, and it will make it slippery so that this can easily move down the gradient. Okay, that is the esophagus during deglutition. This must be clear. And also remember that the glycoproteins of the mucus, they have amphoteric properties. Glycoprotein in mucus that has amphoteric nature. So what does this amphoteric nature mean? That means it is capable of buffering small amount of acid or alkalis. So amphoteric means capability to buffer small amount of acids or alkali. Sometimes we have acid, we take in acidic food or food that is quite alkaline in nature, so that will be there. And also mucus often contains some amount of bicarbonate. So mucus, what is the function of this mucus? So mucus mainly had water electrolytes and glycoproteins. So this mucus also contain bicarbonate ion. So what is the importance of bicarbonate ion? This is to neutralize the acid. Okay. Now moving on to the secretion of saliva. So whenever we are talking about mainly the secretion portion of saliva, remember that saliva has two types of secretion. One is the serous secretion and one is the mucus secretion. Serous secretion, here mainly it will actually secrete alpha amylase or ptyalin, and mucus means that will mainly have this mucin protein present, which is adding this lubrication, making the food particles slippery for easy movement down the esophagus. Now talking about the composition, remember that water is mainly present in saliva. In most body fluids you will find water in higher percentage. Then there are solutes. What are the solutes? They can either be inorganic solutes or there can be organic solutes present. In inorganic solutes, remember mainly potassium and bicarbonate is present in the saliva. More amount is present in the saliva compared to sodium or chloride. This should be very clear, that more amount of potassium and bicarbonate is present in the saliva compared to sodium and chlorine. Okay. Under organic solutes, you have already seen the name. One is alpha amylase, that is ptyalin. There will be mucin, there will be secretory certain IgA lysozymes will be present. Even thiocyanates are also present. These thiocyanates, they are actually antibacterial. Now what is this mechanism of secretion? Now see what will happen. Look at this diagram. What is actually happening here? Constantly, this is the lumen. Okay, this is actually the lumen of saliva. Okay, there are the ducts. You know the salivary glands. There are only three types of salivary gland. One is the parotid gland, one is the submandibular, and one is the sublingual glands. So they have these glands. They are ducts. They have ducts. Okay, so in these ducts, through these ducts, sodium will actually get reabsorbed into the cell. Okay, into the glandular cell, and in that place, potassium will be secreted into the duct. Similarly, chloride will be taken in and bicarbonate will be secreted into the duct. That's why potassium and bicarbonate, they are at a higher concentration in the saliva. So what will happen? There will be actively reabsorbed sodium from the salivary duct, and potassium will be actively secreted in exchange for sodium. So sodium in the saliva, that will reduce, and what will happen? There will be, sodium is decreasing, potassium is coming into the saliva. What will happen? There will be development of minus 70 millivolt in the salivary duct, and when there is already negative, because sodium is at a higher concentration, sodium is moving into the cell, so the ducts, they are having less amount of sodium. So mainly due to potassium's presence, they will actually be slightly negatively charged, because potassium is less present compared to other anions present. And to actually decrease this negativity, what will happen? The chloride which was present, that will be reabsorbed passively, and in that same exchange there will be exchange of bicarbonate. So here bicarbonate will be secreted into the ductal epithelium. Okay, secreted by the ductal epithelium into the duct, and this will further cause chloride to get reabsorbed passively. So this is very important mechanism. If you have seen this diagram, here it is clearly understandable that sodium is getting inside into the cells and potassium is excreted, or rather it is secreted into the saliva. So this will happen. So remember, the concentration of sodium and chloride is approximately 15 milliequivalent per liter in saliva, whereas potassium is approximately 30 milliequivalent, whereas bicarbonate is highest present, that is 50 to 70 milliequivalent per liter. Now coming on to the functions of the saliva, we all know that saliva, it has mainly the lubrication of food, it has mainly adherent to the food particles and all, the mucus present in the saliva. So already we had seen that part. Now most important is that there is presence of this thiocyanate, as I told you, that this thiocyanate is the antibacterial one, and other than that there are lysozymal enzymes also present in the saliva which will kill the bacteria that might have entered with the food. And amylase is there to break down carbohydrate into dextrin and maltose. Some carbohydrates are broken down by salivary alpha amylase or ptyalin. This is the diagram given in Netter's Atlas. So here you can see the location of the glands, the sublingual that is present beneath the tongue, the submandibular duct along with the submandibular gland is present, and this portion is actually the parotid gland. Okay, so parotid gland is present over here. There is the parotid duct or the Stenson's duct from where the secretion will come. Okay, so all the secretion, they are coming into this buccal cavity, and over here only the process is happening, that is the food is actually mixed with the saliva. Now talking about the nervous regulation of saliva, mainly under parasympathetic control, as I already told you. So whenever the taste sensation is coming, how this, let's look at this diagram. So remember, the taste stimuli that is mainly coming from the tongue. So this tongue has two types of taste sensation stimuli, that is, one is the anterior 2/3 and posterior 1/3. So they have different. So posterior 1/3, the taste stimuli is actually sent by the glossopharyngeal nerve, and it is going where? It is going to the superior and the inferior salivatory nuclei, mainly present near the pons or the pontomedullary junction, in that region. Then what is happening over here? Over here the secretion, okay, superior salivatory nuclei, from this secretion via the branch of the facial nerve, okay, that is through the chorda tympani, one side this is going, this is also coming. You can see that from here, the anterior 2/3 of the tongue, this is coming by the chorda tympani branch, facial nerve, and it is going up to here. Then remember that from the superior and inferior salivatory nuclei, what will happen? Via one branch of glossopharyngeal nerve, it will reach this otic ganglia, and from there into the parotid gland, where there will be mainly parasympathetic release of saliva. And another branch from here, this part, you can see that from the salivatory nuclei, here one branch of the facial nerve that is accompanying this chorda tympani nerve, this is coming to the submandibular ganglia, and that is mainly supplying the submandibular gland and the sublingual gland. I'm not going into the details, but remember that facial nerve has separate nuclei. Okay, facial nerve has different nuclei. So let's see once. So facial nerve, if I'm talking about the superior salivatory nuclei of facial nerve. So facial nerve has different nuclei. Of them, superior salivatory nuclei, okay, superior salivatory nucleus that is present, which is mainly bringing the genital, visceral, efferent fibers, okay. And then we have the upper part of the nucleus, tractus solitarius, that is mainly bringing the SV component, that is SV component, or it can also bring the GVA component associated, genital, visceral, afferent component. And what is happening, let me tell you once again, that the anterior and the posterior segments of the tongue, anterior 2/3 and posterior 1/3 segments of the tongue, from there the stimuli, that is the taste stimuli, that is going, see, via posterior third is going via the glossopharyngeal nerve, and it's going to the superior or the inferior salivatory nuclei, and the anterior 2/3, that is via the chorda tympani branch of the facial nerve, that sensory stimulus is also going to the superior and inferior salivatory nucleus, okay, nuclei. And from there, remember that one branch that is accompanying the glossopharyngeal nerve, that branch which is actually controlling the parasympathetic efferent fibers, okay, the parasympathetic efferent fibers, that is going to the otic ganglia, and from there that fibers are going to the parotid gland, from where the saliva secretion is happening. Similarly, from here, another visceral efferent component that is going, that is supplying the parasympathetic, that is coming from the superior and the inferior salivatory nuclei, accompanying the facial nerve and the branch of the chorda tympani branch of facial nerve, and it is going to the submandibular ganglia, from where it is going to the submandibular gland and the sublingual gland. So that parasympathetic control is very important. So whenever you are tasting any food, so you are starting to salivate. Whenever you are thinking of any food, that also you will start salivating. Whenever you are getting the smell of the food, that point of time also you will start salivating, if you know the smell, if your brain recognizes the smell of that particular dish. So there are mainly two types of salivatory nuclei, inferior and superior salivatory nuclei, okay, mainly located at the pontomedullary junction. So they are either they can secrete or inhibit depending on the stimuli, okay, present. So with that, we end our discussion on salivation. We have discussed the functions of salivation, that how it will destroy the bacteria and all, and we have also talked about the nervous regulation of salivation. That was also important. Thank you. Hello everyone, I'm Saptarshi Biswas from Murshidabad Medical College. Last we had discussed about the secretions of saliva. Now we will discuss about the various other secretions of the GIT. Coming on to the esophagus, remember the food bolus, that was after mastication, that is chewing. Then we actually deglutition occurs, that is we are swallowing the food bolus, and along with the saliva mixture, it is coming down the esophagus. Now esophagus has not any other proper type of or specific or specialized glands, only that it has certain compound mucous glands. Okay, it has those mucous glands present located mainly near the esophageogastric junctions, or that region is actually called as the cardiac sphincter. Okay, the stomach has two sphincters on the upper end, that is at the level of the esophageogastric junction it has the cardiac sphincter, and at the level of the duodenum, that is the lower end, it is the pyloric end, it has the pyloric sphincter. So the esophageogastric sphincter mainly it protects the esophageal wall from digestion by the gastric juice whenever we are actually deglutition is happening, so that cardiac openings, cardiac sphincter, that is relaxing and the food is entering into the stomach. So at times there can be reflux of the disease of that gastric juice, like in the case of GERD, gastroesophageal reflux disease, there is already reflux of this and there is peptic ulcer formation in the esophagus. So remember that there is no other, but is the digestion process stopping? No, saliva is present, so saliva will continue its digestion of carbohydrates into dextrin and alpha maltose, that will happen, but of course any other types of digestion will not happen until the food bolus reaches the stomach. So in the gastric secretion, remember, which is produced approximately 1.5 to 2 liters per day, the glands, I'm talking about the glands which are mainly present in the submucosa, that is the oxyntic glands and the pyloric gland. Associated with that, of course, the mucous glands are there. Mucus is very much important. Talking about the oxyntic cells will produce HCl, hydrochloric acid, pepsinogen and caseal intrinsic factor along with some amount of mucus. Where is it located? Remember, the location of oxyntic cell is on the inside portion of the body and fundus region of the stomach. This is very specific because certain regions of the stomach has more number of a particular type of cell. Okay, like the pyloric cells, these pyloric glands, they are only located in the antral region. Okay, now you might not understand, if you have not read the anatomy of stomach, you will not be clear with this thing. So if this is the stomach, okay, like this it has, so stomach has mainly different portions. This portion, this upward portion, this is the fundus. Okay, this is the fundus of the stomach where mainly gas is present, that is called as the fundic gas. Then at this junction, okay, this portion of the stomach, this is called as the cardiac portion. This is the cardiac portion. Then the remaining portion is mainly the, up to this much, this portion is the body of the stomach. Then this portion mainly we find the antrum, and then the lower end of the stomach, that is merging with the duodenum, first part of duodenum, that is basically the pyloric part of the stomach. So as per the location, these glands are also present. Now talking about the mucous gland, it will produce what? The mucus, and automatically this is very important because the mucosa which is there, that stomach layer mucosa, that is constantly being affected by the acids produced from the oxyntic glands. So there is a thick, approximately greater than 1 millimeter thick layer of mucus is present, so that the stomach wall is not auto-digested by its own enzymes and hydrochloric acid. Among these oxyntic cells, there are also enterochromaffin-like cells, ECL cells, which will produce a little bit of histamine, which is basically acting as a vasoconstrictor in most of the cases, or as a vasodilator. It's basically a vasodilator. Okay, serotonin is vasoconstrictor. Mostly, in most of the cases, that's the phenomena. Now see, D cells are also present, which are producing somatostatin. If I show you the diagram, so this is the diagram. Here you see, this basically is the particular shape of the gland. This is the neck portion. Okay. This is the neck portion. There is this MCS, there are the mucous surface cells present. There is the gastric gland, there are the oxyntic cells. Okay, there are the oxyntic cells present. And then other cells are there, like the somatostatin producing B cells are there. Then there are also enterochromaffin like cells, ECL cells. They are present. The mucous neck cells I have already shown you. This is the neck region, so this has this mucous neck cells present. And also there are these parietal cells present, and these are the chief cells. Okay, this CC means these are basically the chief cells. Okay, chief cells are the oxyntic cells which are present. So from here only there is production of HCl. Now coming on to the composition, mainly the stomach juice or any secretion in the body mainly has water in higher percentage, and there are also additional solutes present. Under inorganic portions, we know that HCl, which is most important, and other than that, other ions like sodium, potassium, bicarbonate, these are present. Whereas under organic solutes, there can be this inactivated enzyme pepsinogen that is released from the peptic cells. The pyloric cells or the peptic cells, they are producing the pepsinogen. Okay, then we have gastric lipase. Remember, a very small amount of lipids, they are also digested in the stomach. Most lipids, they are requiring emulsification before digestion and before absorption. Okay, that is happening in the intestine, small intestine. But a little portion of already emulsified fat, certain fat substances, if they are already broken into droplets or if they are smaller in size, those fat droplets, they are basically digested by gastric lipase, mainly producing triglycerides and fatty acids. There is also production of intrinsic factor, very important for absorption of vitamin B12 from the ileum, mainly from the ileum. This requires this caseal intrinsic factor. Okay, when we actually discuss anemia, megaloblastic anemia, that point of time we talk about this particular, that there are other substances which are added to the intrinsic factor, and the intrinsic factor actually enters into the cells of the ileum. Here I am not going into those details. Now mainly talking about the mechanism of formation of HCl. Now see, you should know that whenever it's stimulated, the parietal cells, they will produce this acid solution, which is approximately around 160 millimole per liter of hydrochloric acid. So what is the secretion amount? That is, what is this amount? 160 millimole per liter of HCl is produced. Okay, which is nearly isotonic with the body fluid. But the pH, thus the pH, there is, of course it has a range, but approximately it is about 0.8, demonstrating its extreme acidity. But yes, the range can be approximately about 1 to 2, we take the approximation, but here in Guyton, in some part it is written that it is 0.8. Then at this pH, what will happen? The H plus ions concentration will be, of course, very, very much higher than what is present in the arterial blood. This is very clear. And what will happen? there is of course requirement of tremendous amount of energy to concentrate the H plus. Okay, the concentration is happening, so there is requirement of tremendous amount of energy. At that same point of time, when the H plus they are secreted, H3O plus will diffuse into the blood, so that the gastric venous blood that has a higher pH, that is, it is becoming alkaline compared to the arterial blood. So the arteries which are supplying, they are having higher concentration of H plus. Are they having? So from there, what is happening? H plus is going and it is producing what? It is producing the HCl. So gastric juice has very much higher H plus compared to the arterial blood. But whenever in the venous blood, remember, in the venous blood there is high percentage of bicarbonate, which is giving that alkaline nature to the venous blood, that is stomach is from the stomach which is coming out. Okay, and as I've shown you over there, in that there, in the oxidative of the parietal cells, oxygenating of the parietal cells, there were presence of this canaliculi. Remember one point. One point must be very clear now. Starting with the remember that the main driving force for HCl production, what is this driving force? This is mainly contributed by hydrogen potassium pump. Okay, and that is actually requiring ATP, so that is an ATPase pump. So this is very clear. So what will happen actually? So this is the parietal cell, this is the lumen of the canaliculi, and that's the interstitium. So water inside the parietal cells becomes dissociated into H plus and OH minus. So water present inside, that will automatically dissociate into H plus and OH minus. So water which was present over here, this water is constantly via osmosis, this is moving into the parietal cells and eventually into the lumen, because lumen of the canaliculi that has less water, so water always follows this passage, that is, it is having passive diffusion via osmosis. So osmosis is happening, so it is coming from the interstitium to the lumen of the canaliculi. So what will happen over here? The H plus which actually broken into H plus and OH minus in the cell. So some amount of water also from here that got broken into H plus and OH minus, that is dissociation. And the H plus is then actively secreted into the canaliculi in exchange of potassium. So this is coming out. Okay, H plus is coming out, and in this exchange potassium is entering. So potassium is entering into the cell. So what is present over here? Here there is presence of the exchanger, that is H plus K plus ATPase pump. So here hydrolysis of ATP is happening, and from here what is happening? Potassium is entering into the cell. Okay, and K plus H plus is going into the lumen. Now what will happen on the basolateral side? What is happening? The basolateral side, there is activity. Always remember, towards the interstitium there is activity of the sodium potassium ATPase pump, that we had already seen in the renal physiology chapter. What had happened over there on the basolateral side, of the side that is facing the interstitium, there is a presence of sodium potassium ATPase pump. So what will happen? There will be movement. So potassium ions are transported into the cell by sodium potassium ATPase pump on the basolateral side and tend to leak into the lumen, but are recycled back by the hydrogen potassium. So potassium leak out. Okay, potassium wants to leak out to the lumen, but what will happen here? Potassium will be again taken in. So what is happening over here? So this potassium which is coming inside, this potassium has a tendency that, what will this potassium do? This potassium is actually coming from this side. Okay, and in this exchange there is sodium is coming out into the interstitium. So here there is presence of this pump, sodium potassium ATPase pump. So this is requiring ATPase. Okay, and this sodium which is mainly present over here, this side of the sodium which is mainly having almost about 3 milliequivalent per liter, this is entering into the cell. Here there is higher concentration of sodium, and sodium is moving eventually out, and potassium in its exchange in presence of ATPase pump is entering the cell, and potassium has tendency to move out of the cell also. Potassium is tending to move out of the cell, and again potassium is recycled back using this hydrogen potassium ATPase pump. This must be very much clear to you. Now talking about in the interstitial fluid. In the interstitial fluid, there is presence of carbon dioxide, because carbon dioxide is produced. So carbon dioxide is present, so this carbon dioxide will actually cross the membrane easily, and carbon dioxide will enter into this cell. Carbon dioxide in presence of water in the cell, it will, in the presence of carbonic anhydrase enzyme, it will form carbonic acid. Now carbonic acid will split into H plus and HCO3 minus. Okay, so it will split into H plus and HCO3 minus. So what will happen? This H plus which is produced, so this H plus has had tendency of moving out, so H plus will again move out. Okay, H plus will move out. So this part should be very clear, that how H plus is moving. Here water is dissociating, from there what H plus is moving into the lumen, and at the same point of time carbon dioxide which is forming carbonic acid and all, that is also moving. Now this bicarbonate which is produced, this will actually be exchanged for chlorine from the interstitium, that will come into the parietal cells. So here there is basically presence of chloride bicarbonate exchanger protein. Okay, there is presence of chloride bicarbonate exchanger protein, and chloride is coming inside. Now this chloride will actually move into its further concentration, that is, this chloride will seem to move out. Okay, so H plus was here, chloride was here, so this will actually form the HCl. Okay, and already remember, some amount of chlorine is actually directly moving through chloride channels. There are chloride channels present which are directly moving this chloride, okay, through the cell into here, because of the gradient difference. There is a gradient difference, so chloride will automatically move. So pumping of H plus and out of the cell by hydrogen potassium ATPase permits hydroxyl ions to accumulate and form bicarbonic bicarbonate to form carbon dioxide. So this carbon dioxide is also, this OH which was present, this OH was also converting and adding and forming bicarbonate, which is exchanged. So this OH which is present, it is coming and contacting with this bicarbonate, OH minus and bicarbonate. Okay, this bicarbonate, this is mainly in presence of carbon dioxide. This is producing this bicarbonate, and this bicarbonate is exchanged for chloride. Okay, there is this exchanger. So chloride is entering into the cell. So this must be very clear that bicarbonate is then transported across the basolateral membrane into this extracellular fluid compartment in exchange for chloride, which is secreted via this chloride channel into the canaliculi, giving a solution of HCl. Okay, so mainly you can see the ions which are present at higher concentration, which is H plus and Cl minus, that will be present. Water automatically by osmosis moves from the interstitium into the lumen, so that I've already discussed. So about 150 to 160. Here I've written approximately, remember this is the value, but there is always a range, so it is 150 to 160 milli equivalent per liter of HCl that is produced. Okay, and potassium chloride also. So this potassium which was actually coming to some extent, okay, this potassium which was coming to some extent, so this potassium will also, in the presence of this chloride, they will also form potassium chloride. Okay, and this potassium chloride will be approximately at a concentration of 15 milli equivalent per liter, and if some amount of sodium is there, then sodium chloride will also be formed. Now this is very important, that how the regulation is happening. Now this is very important, that is production of HCl. Okay, now coming on to the regulation of the gastric acid secretion, that is mainly controlled by certain factors like their acetylcholine, gastrin and histamine. Now talking about first acetylcholine. Acetylcholine, which is released from the parasympathetic stimulation, will excite secretion of pepsinogen by the pepsin, that is by the peptic cells. So what will happen? Acetylcholine, that will stimulate the peptic cells, and what will happen? Pepsinogen will be produced. So pepsinogen is important protein digesting enzyme. This is a zymogen or a pre-enzyme that will be converted into pepsin. Both gastrin and histamine, they will also, what will happen? Hydrochloric acid by parietal cells and mucus by. So pepsinogen is produced by the peptic cells from the oxyntic cells. This ACH will stimulate, and there will be production of HCl. Okay. Similarly, what will happen? Parietal cells will also close association with enterochromaffin-like cells, which will actually produce histamine. So this will, therefore, the release histamine will actually also stimulate secretion of acid. So histamine, which is produced from the enterochromaffin cell, that will also stimulate oxyntic cells for HCl production. Similarly, the gastrin is a local hormone produced by the G cells. Where are the G cells present? They are mainly presented in the close to the pyloric glands, mainly in the antral portion. So that will also stimulate HCl production. So gastrin, histamine and acetylcholine, all these, they are basically stimulating HCl production from the oxyntic cells. This gastrin is actually a large polypeptide secreted in two forms. The larger form, gastrin has, remember this is a large polypeptide. So gastrin has mainly two forms. Larger form is actually G34 and smaller form is basically G17. So remember, this thing is very important. Now coming on to the phases of gastric secretion, mainly remember there are three phases. One is the cephalic phase, one is the gastric phase, and one is the intestinal phase. So cephalic phase, what is happening? Because even before, when the food is entering the stomach, especially when it's been eaten, that is whenever you're chewing the food, at that point of time, or it can happen at the time of sight or smell or taste of the food, or you're thinking of that food. So automatically in this phase, there will be 30% of the gastric secretion. Regulation, the neurogenic signals will actually cause the cephalic phase. That means here you're thinking. So there will be involvement of the cerebral cortex and the appetite centers of the amygdala and the hypothalamus. So these stimulations by the dorsal motor nuclei of vagus nerve, and they will reach the stomach for its secretion. Next is the gastric phase. What will happen whenever the food is entering into the stomach? That is when digestion happens, deglutition happens, and food enters into the stomach. At that point of time, gastric juice will be produced. Okay, and remember here it will remain for several hours. This food will remain in the stomach, this churning will happen, and in this phase there will be about 60% release of gastric secretion. 60% will be released. This phase is mainly regulated by the long vagovagal reflexes, the local enteric reflexes, and again by the gastrin, histamine. These local hormones will also play an important role. Next is the intestinal phase, when the food will enter into the upper portion of the small intestine, that is mainly in the duodenum and the jejunum portions partly. That time a little portion, that is about 10% of acid, will also be secreted, and this is based on the hormones that are released, the local hormones, and also to some extent on the nervous system. So these phases of gastric secretion is very important, and there will also be at times inhibition of gastric secretions by other intestinal factors. The presence of food in the small intestine will initiate a reverse enterogastric. I'm writing over here. What will happen? Inhibition of gastric secretion. Inhibition of gastric secretion. How will it be inhibited? We had discussed about the stimulating factors acetylcholine, then gastrin and histamine. Now talking about the inhibiting factors, whenever there will be presence of food, very common, when there will be presence of food in the intestine. Okay, so this will actually stimulate. What this will stimulate? This will initiate a reverse. This will stimulate a reverse enterogastric reflex. And this will be transmitted to the myotonic nervous system and extrinsic sympathetic and vagus nerve, and automatically via vagus, then myenteric system, and there will be extrinsic sympathetic system, what will happen? There will be inhibition of stomach secretion. Okay, there will be inhibition of stomach secretion. That can happen due to distension in the bowel, small bowel, or presence of acid in the upper portion of the intestine, or presence of protein breakdown products over there. The presence of acid fat proteins. Okay, these hyperosmotic and hypoosmotic fluids, they cause irritating factors in mainly in the upper smaller intestine portions, and there will be release of hormones like secretin, which is controlling the pancreatic secretion. So this secretin will oppose stomach secretion. This is very important. If they ask you about a very specific local hormone that will inhibition, so remember the action of secretin. So secretin will say stop. It will say stoppage to the gastric secretion, and it will stimulate what? It will stimulate the pancreatic secretion. So secretin will oppose the stomach secretion. The other hormones can be glucose dependent insulinotropic peptide, that is gastric inhibitory peptide. So actually it is now called as glucose dependent insulinotropic peptide, okay, GIP. And there is vasoactive intestinal polypeptide, VIP, and there is somatostatin. Okay, they actually, what they do, they all inhibit the stomach secretion. Secretin, GIP, VIP, somatostatin, they have basically this phase. Now remember gastric secretion, what will happen during the interdigestive phase. So here I've discussed. Now what will happen, mainly gastric secretion in interdigestive phase. The stomach secretes a few millimeters of gastric acid each hour. During this interdigestive phase, the secretion that occurs mainly is non-oxyntic type, and it has mainly mucus and little amount of pepsin and almost very little or no amount of acid. Okay, so whenever there is emotional stimuli, this can increase the interdigestive gastric secretion, which is highly peptic and acidic, to 50 ml or more per hour. That is more secretion, that is the cephalic or the gastric secretion, that will happen more compared to the onset of a meal. This increase of secretion in response to emotional stimuli may contribute to development of peptic ulcer at times. So what will happen in the interdigestive phase? What can happen if there is emotional stimuli? So generally in this stage, little amount of acid is produced, but whenever in cases of emotional stimuli, that will cause excess amount of production of gastric secretion. So here this will produce gastric secretion, and remember this secretion has highly peptic, that is pepsinogen present, and acidic. That means actually increase more than 50 ml secretion per hour. So this will be same way, that is the cephalic or the gastric phases, they will actually be comparatively equal, or at times it can be more, okay, compared to what amount of gastric juice is released in the cephalic or in the gastric phases. Okay, so this can actually lead to peptic ulcer. This is very important. Another important point you should remember, that whenever this HCl and pepsin, they are coming, so HCl, whenever it is coming in contact with pepsinogen, this is actually turning or activating it. This is converting it into pepsin. Okay, pepsin is this activated product, active pepsin. This is actually, this pepsinogen is actually a pro molecule, and now pepsin is basically a comparatively lighter molecule also, because certain proteolytic activity is there, and it is actually operating, this pepsin is protein digesting enzyme, which is operating at pH range of approximately 1.8 to 3.5. In this range, what will happen? This is basically a proteolytic enzyme. So whenever it's a proteolytic enzyme, this will actually start digesting the proteins. So the proteins which have come into the food bolus, they will be digested by pepsin, and those proteins will be broken down into peptide chains. They can be oligopeptides, or there can be some oligopeptides can further be broken down into amino acids. Now intrinsic factor I've discussed, that was essential for vitamin B12 absorption from the ileum. Now coming on to the regulation of pepsinogen. So acetylcholine, that is released from the vagus nerve, that is parasympathetic nervous system, and the acid in the stomach. So acid probably does not stimulate the peptic cells directly, but instead they will elicit additional enteric neural reflexes that will actually support the original nervous system, and therefore rate of pepsinogen will actually increase. So when pepsinogen is increasing, automatically HCl will convert them into pepsin. So this must be clear. Then other molecules are there, like gastrin, cholecystokinin, and secretin. These are actually larger polypeptides. Okay, they have higher molecular weights. So they have specific functions also, and one of a synthetic gastrin. We were discussing about this gastrin hormone. So this gastrin local hormone has actually a synthetic form that was developed, synthetic form, and that synthetic form is actually called as pentagastrin, which is composed of terminal 4 amino acids of natural gastrin, plus there is an additional amino acid alanine. Okay, it has similar properties to that of natural gastrin, only remember it's a synthetic form, so it has terminal 4 amino acid, plus additionally it has a alanine present. Okay, so it has same, so pentagastrin is one of them. Now talking about the major GI hormones, some of them we have discussed. First is the gastrin from the G cells of the antrum of the stomach, that is increasing the gastric acidic content, and it's also lowering the GI motility. Okay, because in the gastrin that will help in HCl production. HCl is required, so churning of food will happen inside the stomach. So that means it is increasing the motility or movement. Okay, so that food remains in the stomach for more duration of time, and that is churned properly by HCl. Secretin, which is from the S cells of the duodenum, that is mainly acting on the acidic chyme. Now the substance that is released as a thick acidic paste from the stomach, after certain time durations, the pyloric end of the stomach will open, and that will facilitate the movement of this thing, that is this acidic chyme is released from the stomach slowly in small amount into the intestine. Okay, so secretin is mainly acting over there, and it will increase the pancreatic buffer bicarbonate secretion. It will decrease gastric gastrin, it will decrease the action of gastrin that I have told you, and that is also decreasing gastric emptying, because if more amount of food enters, then automatically the acidic chyme's function is to stimulate the pancreatic secretions. Now if more and more acidic chyme is entering, so there will be a problem on the pancreas also, so secretin will actually say stop to the acidic chyme, and it will only allow secretions from the pancreas. Now CCK, that is actually called as CCK-PZ, cholecystokinin pancreozymin, from the eye cells of the duodenum and jejunum, they are also acting, they're mainly acting on the small peptides and amino acid fats. They're also increasing pancreatic and biliary buffer secretion. Okay, just remember some main functions. Talking about the gastric inhibitory peptide, also called as the GIP. So that is mainly produced from the duodenum and jejunum, that is causing increase in gastric secretion, and that is delaying gastric emptying. Motilin and peptide YY dash, so that is released from the M cells of the duodenum and L cells of the intestine, and they are mainly happening in the phase 3 contraction of the migrating motor complex, MMC. Okay, I will discuss sometimes later. In the next part, I will discuss about the migrating motor complex, but remember that is very important. Migrating motor complex, that is production of motilin is happening. Whenever there is no food in that serving stages, that time motilin is actually produced. That is mainly called as and development of certain impulses. That is the migrating motor complex that I will discuss later. So with this, we have ended gastric acid production. A small topic is left, that is migrating motor complex, which I will discuss in the next video. Thank you. Hello everyone, I am Saptarshi Biswas from Murshidabad Medical College. Now we will be discussing about the gastrointestinal motility. Now whenever we are taking in the food, that food is passing down into the stomach via the esophagus. From there, the contents, they are digested in the intestine, their digestion is happening. From the intestine, absorption is happening, and that particular nutrients, they passed into the liver via the portal circulation. So their assimilation is happening, and then whatever undigested portions are remaining in that food, they are excreted. So this requires motility, because our GI tract is very long, because of the small intestine, which is highly coiled, the large intestine, which is coiled and present inside our abdominal region. So that is actually quite long. So this entire length of the GIT has to be traversed by the food particles, and after that the unwanted products, they have to pass. So that requires continuous movement. Okay, so the functional movement, there are two types. One is the propulsive movement and one is the mixing movement. So propulsive movement, they are the peristaltic movements where food moves forward along the GIT at a certain rate. That depends on the rate of peristalsis. And one is the movement of mixing that mainly happens in the stomach, where the food contents are thoroughly mixed and churning of food occurs in the stomach. Now what is this peristalsis or propulsive movement? Whenever, like at time 0, let's see that in this case here, a particular segment, this segment has contracted, and after 5 seconds we see that the contraction, this has shifted here. So there is certain shift. Okay, one portion of the tube is constricting, the muscles are constricting, and another portion is relaxing. Then the constriction moves forward, and then that forward region gets constricted, and the remaining regions, they are remaining in the dilated version. So that happens, and that is an inherent property of the syncytial smooth muscles. That is for the GIT muscles also this is applicable, because they are also syncytial smooth muscles, and they also occur in certain ducts like the glandular duct, the bile ducts, or the ureters. What are the factors that will cause peristalsis? There is gut distension, either due to flatulence. There can be, like in the fundus, if there is gas, so that can also cause certain amount of distension if there is increase in the fundic gas, or if there is excess amount of food in the stomach, that can also cause GI distension. So this can happen if there is any sort of physical or chemical irritation happening in the gut epithelium due to any type of chemical factors or physical factors. So that can also increase peristalsis, and there can be strong parasympathetic stimulation which can increase the peristalsis. Now see the meyenteric plexus. This is the plexus or the Auerbach's plexus. This is present between the longitudinal and the circularis muscles of the muscularis externa. So these are very important, because these muscles are actually constricting. There is contraction of the muscles of the circularis muscles and the longitudinal muscles, and especially in the stomach we have the oblique muscles also. So they are contraction is happening and relaxation is happening. So that is actually causing this propulsive movement. But if there is congenital absence of the meyenteric plexus, then you will know the peristalsis will actually be weak and may not happen in certain portions of the GIT. Or like if there is, you have taken, there is atropine, if there is excess dosage of atropine, then that will also inhibit the cholinergic nerve endings of the meyenteric plexus, leading to this weak peristalsis. Okay, now peristaltic waves move towards anus with downstream receptive relaxation. That is the law of gut. Now let me explain what is this law of gut. Peristalsis, theoretically it is said that it is starting from at this point and it is moving in both directions. That is, this is the anal side, this is the mouth side. But it is said that when this is simulated at one point and it is moving towards the mouth, then what will happen? This peristalsis will actually die out. This particular portion will die out, and the remaining portion, this portion that is the movement towards the anus, that will actually continue. So peristalsis will continue towards the anus. That is, it will be a directional transmission of peristalsis. Okay, when the segment of intestinal tract becomes excited by distension and thereby there is initiation of peristalsis, the contractile ring causes this segment and moves towards the distended segment, pushing the intestinal component towards the anal direction. Okay, and that is happening. At the same time, the gut sometimes relaxes also several centimeters, that is downstream also towards the anus. There is also receptive relaxation happening. So food is propelled more easily towards the anus than towards the mouth. One side, this portion is dying out. Now let me write, when segment of intestine is excited, at that point of time what is happening? This will initiate peristalsis. Okay, and what will happen? There will be contractile ring causing peristalsis normally begins. So this contractile ring, that is the muscles, causes peristalsis. It will originate from basically the upper portion, that is from direction of towards the mouth. From that region it will originate. Okay, and move towards the lower segments, move towards lower part of GIT, And when it will move to the lower part of the GIT towards the anal direction, what will happen? Propel intestinal contents. There will be propulsion of the intestinal contents, okay, in anal direction. At same time, what will happen? The gut will relax. Gut at certain places will relax also. Where will it relax? That will be several centimeters down this. If suppose one portion is having certain amount of, if one portion is having this constriction, okay, then what will happen? Several centimeter downwards, this will be in which state? Okay, in this portion, this will be in that relaxation phase. Again, when this will move to this portion, this will again constrict like this. Okay, so that is happening constantly. So this is actually explained over here. That is called as receptive relaxation downstream. And sometimes the gut sometimes relax several centimeters downstream, and this is called as receptive relaxation. And that actually facilitates movement of the contents towards the anal side. Okay, so this complex pattern does not occur in the absence of mesenteric plexus, because mesenteric plexus are coldly involved in this. This is called as the mesenteric reflex or peristaltic reflex. Okay, so this receptive, this is called as mesenteric or peristaltic reflex. Okay, and this peristaltic reflex plus anal direction movement, this is called as law of gut. So peristaltic movement plus anal direction movement, that is movement towards the anus, that is together called as the law of gut. Okay, now processes that are occurring in the GIT, we are all familiar with the processes. First is the ingestion of the food, then we have digestion, then we have absorption, assimilation and excretion. Now see, when we are talking about ingestion of food, that is happening mainly, first there is mastication. Mastication means chewing. We are chewing the food particles which are comparatively of a larger size, so they are chewed. Now muscles of mastication which are present, they are controlled by the motor branch of the cranial nerve 5, and chewing, that is mainly controlled by certain nuclei in the brainstem. I'm not going into the muscles of mastication, their nerve supply, in details in anatomy they are discussed. Now talking about the chewing reflex, that what is happening in this chewing reflex, let's see. So there will be presence of food bolus in mouth. Okay, so that will cause reflex inhibition. When this is present, this will cause reflex inhibition of muscles of mastication. So what will happen? This will cause the lower jaws to drop, because that is a proper synovial movable joint present in the skull region, that is this lower jaw movement, temporomandibular joint. So that will move, and this will cause what? This will initiate a stretch reflex. This will initiate a stretch reflex, which will further cause rebound contraction, and that will cause raising of jaw and closure of teeth. That is happening simultaneously. You can understand that when you are chewing the food, what are the steps that are happening. So you can easily imagine that thing, and this will compress the bolus again against the oral lining. So compression of bolus against oral lining. So that is basically chewing reflex. You are chewing now. Inside, what is happening? There is food bolus. So muscles of mastication, first there will be reflex inhibition, the lower jaw will drop, and that will again initiate a stretch reflex, and there will be again rebound contraction. So continuously you are chewing the food. It's not like that you are once chewing and letting it go. No, you are constantly chewing the food. There will be rebound contractions, and then again the raising of jaw and closure of teeth will happen. You will again chew, and this will further compress the food bolus against the oral lining, and there will be proper mastication. Now talking about swallowing or deglutition, this has mainly 3 phases. Swallowing, then you will swallow the food. One is the voluntary stage. You will push the food bolus with your tongue into the back on the posterior mouth, and then automatically into the oropharynx. Okay, so there will be voluntary squeezing or roll posteriorly into the pharynx by the tongue, and you will press it of course against the palate, and then you will move it automatically. Your tongue muscles will play an important role, and you will actually complete the first phase, that is voluntary stage of deglutition. Then coming on to the involuntary pharyngeal stage, when it enters the posterior mouth and pharynx, that will stimulate this epithelial swallowing receptor areas all around that opening of the pharynx, and mostly on the tonsillar pillars, and that impulses will be sent to the brainstem, that will initiate the automatic pharyngeal muscle contraction. Whenever that portion will reach your pharynx, there are certain receptors present, epithelial swallowing receptors, that will stimulate impulses, send impulses to the brainstem, and they will stimulate the automatic pharyngeal muscle contraction over there. Then soft palate is pulled upward. This is important. The soft palate, that portion, that is the uvula, that is basically present as a continuation of the posterior part of the soft palate. That portion will be pulled upwards, and that will close the posterior nasal nares, so that the food particles cannot enter into the nasal cavity, okay, into the posterior nasal cavity. So that is prevented by this soft palate, velum palatii or uvula. Okay, then palatopharyngeal folds are there. Now about these regions, the muscles present around this, the stylopharyngeus, the glossopharyngeus, all these different muscles are there. Muscles of mastication, muscles of deglutition, these are well discussed in anatomy, so I'm not going into those details. If you have read that portion, you will be you can relate it easily, that the palatopharyngeal folds are present on each side of the pharynx, and they're pulled medially into the sagittal sling, and that the food passes mainly into the posterior pharynx, that is in the region of oropharynx. Okay, and automatically from oropharynx it will pass to laryngopharynx, because already the food did not go to the level of the nasopharynx. It is mainly coming to the region of oropharynx, and then it will go to laryngopharynx. So larynx is pulled upwards and anteriorly by the neck muscles. So which muscles are playing important role, you know very well. And what will happen, the epiglottis will move backward, and that will close the opening of the larynx into the trachea. So that region should be blocked, because you know epiglottis is that muscular fold which is present on it, and that fold should be closed. Okay, epiglottis, that fold closes the trachea so that the food does not enter into the trachea or the windpipe. And this is provided, this tight approximation is provided by the vocal cords. Upward movement of the larynx, that will enlarge the opening of the esophagus. So that will happen, of course, because larynx is also moving associated or accompanied by the movement of muscles of the neck. So they will actually cause enlarged opening of the esophagus, and food will move inside. Relaxation of the upper esophageal sphincter will happen, that will cause propulsion of the food down the esophagus. And time is taking approximately, somewhere it's in less than 6 seconds, someone is saying less than 2 seconds. So remember that passage of food through this upper esophageal sphincter, that will take time less than 2 seconds. Okay, whereas the pharyngeal stage of swelling, that occurs mainly in time duration of less than 6 seconds. And during this phase, remember, only a portion of respiration or respiratory cycle is interrupted, because when epiglottis is closed during deglutition, that time you cannot exchange air. So the mechanical phase of respiration is closed. And swallowing center actually inhibits when there is, you have seen that epithelial swallowing centers present around that constricted regions of the pharynx. So that will again inhibit the respiratory centers of the medulla, so that at that point of time there is no exchange of gases generally. Okay, so that control is maintained by the body. Now talking about the nervous initiation, the sensitive tactile areas of the posterior mouth lying and around the pharyngeal opening, this I have already discussed. That impulse will actually be transmitted via the trigeminal nerve and the glossopharyngeal nerve. Okay, and that will reach the medulla oblongata, which will be in close association with the nucleus tractus solitarius. So that was about the sensory impulse that was moving via the two nerves, that is trigeminal and glossopharyngeal, their sensory portions or their sensory afferents. Then talking about the motor impulse from the swallowing center, the motor impulse will actually be sent by the cranial nerve 5, 9, 10 and 12. Okay, and that will actually cause impulses will be coming to the pharynx, where the muscles of the pharynx, they will basically be contracted, and relaxation, all this control will be done. Now talking about the last stage, that is the esophageal stage of swallowing. Here peristalsis happens in this phase, and there are two types. One is the primary and one is the secondary peristalsis. Primary peristalsis will force the movement of the food or food bolus from pharynx to esophagus and then to stomach, which will approximately take 8 to 10 seconds time. Generally it's less than 8 seconds. And in the secondary peristalsis, what will happen, this will result from the distension of the esophagus by the retained food. Of course, esophagus, that is not so thick structure, but food bolus is passing down, so there will be distension of the esophagus, and this intrinsic neural circuits of the mindfree plexus will get activated. Okay, there will be activation, and this activation will send efferents to the medulla via the vagus afferents, and again to the esophagus, that will be sent back by the cranial nerves 9 and 10. These are the efferent nerve fibers of the cranial nerves 9 and 10. So they will again send back the message or the motor impulse back to the esophagus like this. So control of the esophageal muscles, upper 1/3 is basically the skeletal muscles or striated muscles, which are mainly controlled by the cranial nerves 9 and 10 efferent nerve fibers, whereas the lower 2/3 segment is basically under control of the vagus nerve, which has basically smooth muscles. This should be very clear, that esophageal muscles, their extent. Okay, now talking about the stomach. Now see, this is the diagram, stomach. So stomach has this region, that is a constricted portion. This region is actually called as the lower gastroesophageal sphincter. Okay, and that is also called as the cardiac sphincter, which is 3 cm above the junction with stomach. Okay, almost 3 cm. So this region is basically the cardiac sphincter. This portion of the stomach, we already know, this is basically the fundus, where fundic gas is stored. And then this is main region is the body, lower portion, this is basically the angular notch. Here actually we find the presence of antrum. Okay, this region is called as the antrum. And then we have the pylorus, which has this guarded by the pyloric sphincter. So these sphincters are basically circular muscles which are covering that opening. Okay, and whenever food will come, that will stimulate, and there will be what? They are already in the constricted phase. There will be relaxation and opening of the or dilatation of this sphincter, so that the food will enter, or from here the food will exit into the duodenum. Okay, so this must be very clear. Function of the lower esophageal sphincter, this is important, which has circular muscles and normally remains tonically constricted due to that intraluminal pressure that develops inside the lumen of approximately 30 millimeters of mercury. Now, what can be the pathological scenario if there is any type of defect or inability of the sphincters to relax when the food is coming? That condition is called as eclasia cardia, and that is very difficult, because you will have problems in engulfing the food. You will take the food, but if that sphincter is not working, so food will start accumulating in the esophagus. That will cause immense amount of irritation, and that can even cause you to vomit. Okay, so that time you will have difficulty in consuming food. So that is a condition, rare condition, eclasia cardia, or it can be even stimulated if the sphincters are not working due to any sort of nervous defect that is not happening. So that can also be there. Now, prevention of the esophageal reflux by valve-like closure of the distal end of the esophagus. So that esophageal reflux is actually prevented. If anything is gastric reflux, is also prevented by the cardiac sphincter. Prevention of the esophageal reflux by valve-like closure of the distal end of the esophagus. What actually happens, that this esophageal end slightly goes into the esophageal portion, slightly extends into the stomach. Okay, at inner point. So if there is intra-abdominal pressure, what will happen? This portion will actually, this inward point, this will actually slightly extend to the stomach. Already this portion will get constricted. Okay, and that will act like a valve-like closure. What are the motor functions of the stomach? Motor functions of the stomach, one is the storage activity. They will store it for longer durations, approximately 5 to 6 hours. The food is stored in the stomach, where churning of the food happens, mixing with gastric juice, and then there is the formation of the chyme, and after that there is slow emptying of the stomach. Now, food mixing and propulsive movement in the stomach, that is maintenance of this basic electric rhythm or basal electric rhythm. Basic or basal electric rhythm, what is that? If there is food in the stomach, there will be weak peristaltic constriction waves. These are called as the mixing waves. Okay, if that is present, that is basically okay. So there will be weak peristaltic constricted areas which will have mixing waves generating, because these are weak peristaltic contractions, and they will mainly happen in the mid to upper portions of the stomach wall and eventually move to the antrum in every 15 to 20 seconds. So this generation of this constriction, peristaltic constriction at the upper levels of the stomach and slowly progressing into the lower level, that is called as the basic electric rhythm. And these waves will intensify on moving down towards the antrum, and these powerful peristaltic action potential driven constrictor rings, they will force the antral contents under higher pressure to the pylorus, and then in the pylorus it will to, and slowly from the pylorus there will be release, that is stomach emptying. Now higher contractions, if there is hunger contractions, when the stomach is already empty for several hours, so there is sometimes you feel mild pain. Okay, that they are called as the hunger pangs. Okay, your stomach is empty, so you need food. That point of time hunger pangs actually happen. Now talking about gastric emptying or stomach emptying, there will be intense antral peristaltic contraction. Those contractions, those BER which were generating in the upper ends, they are coming downwards, and they will intensify and create pressures of about 50 to 70 centimeter of water, which are 6 times more powerful. Okay, and these will cause several millimeters of chyme movement into the duodenum. Slowly the pyloric sphincter, the circular muscles will relax, and the food will move from the duodenum into the from the pylorus into the duodenum. What is the role of the pylorus? The pylorus will be slightly tonically contracted by the circular muscles. This sphincter will be formed, so that is the role of pylorus. Now certain hormones, local hormones which are responsible for gastric emptying, one is the food volume. First normal case, that is food volume. You know that if the food volume is more, so there will be stretching of the stomach wall, and that will cause the local mesenteric reflexes, and there will be faster emptying. Due to action of gastrin, gastrin always increases gastric motility, and the motor function of the stomach will be increased, activity of the pyloric pump will increase, so that will cause increased gastric emptying. Duodenal factors, certain duodenal factors will inhibit gastric emptying, because when the food is coming to the duodenum, duodenum needs to digest the food properly and further send it to the jejunum and the ileum. So there you cannot send extra amount of food or chyme. Basically that acidic paste is the chyme that is sent, so that will not happen. So duodenal factors will inhibit the gastric emptying, like they have the enterogastric neural reflexes. So that can call directly from the duodenum, they can go via the enteric nervous system, or certain intrinsic nerves are there which will carry this efferents to the this afferents will be carried to the prevertebral sympathetic ganglia, and inhibitory sympathetic fibers will actually cause them. This is going, efferents are going to the prevertebral ganglia, sympathetic ganglia. From there, the inhibitory efferent signals are coming to the stomach. So that can cause stomach walls to actually not release. The pylorus will not relax at that point of time. Or there can be the vagus nerve stimulation, which will go to the brainstem and inhibit the normal excitatory signals to the stomach. This should be clear. Now, other hormones, local hormones, there we have discussed gastrin, which was stimulating gastric motility. Here we have CCK-PZ, that is cholecystokinin pancreozymin, which is released from the mucosa of jejunum, that will actually inhibit or reduce stomach or gastric motility. So thereby inhibiting or reducing gastric emptying. Okay, secretin and glucagon, that is gastric inhibitory peptide. Also we call it as glucose dependent insulinotropic peptide. So that will basically inhibit gastric motility. Now talking about a very important topic, that is migrating motor complex. Now, what is this migratory motor complex? Let's see. So it's a recurring cyclical contractions that occur in the stomach and small intestine in the fasting or interdigestive phase. So in fasting or interdigestive phase, what is happening? There is recurring cyclical contractions, cyclical contractions of stomach and small intestine. So that is called as migrating motor complex. Okay, MMC. So motilin, that is basically a gastrointestinal hormone. So two hormones are responsible. One is motilin and another is serotonin, that is 5-hydroxytryptamine. Okay, serotonin and motilin, these two hormones are responsible for influence of MMC. This will influence or stimulate migrating motor complex, and they will send a positive feedback to the stomach and small intestine. Now see, there are phases. First is the phase number 1, which has no visible contraction or quiet period, that is phase 1. Phase 2, here low amplitude contractions are happening. In phase 3, there is higher recurring high amplitude contractions, and in the phase 4, there is basically transition from phase 3 to phase 1, that cyclical process, because you have written over here, this is cyclical. So this process starting from 1, it is going to 2, most intensified is 3, and this process of back from 3 to 1, that is called as the 4th phase. Okay, so motilin and serotonin, these are important over here. Phase 3 contractions in the stomach are mainly influenced by motilin secretion, that is mediated by vagus nerve. So motilin is actually initiated by vagus nerve or mediated by vagus nerve. Phase 3 contractions in the small intestine are not affected by vagus nerve. So this happens only in the stomach, not for small intestine. Remember this point. Okay, that only for the stomach, the vagus nerve will actually mediate the motilin and they will cause migrating motor complexes, but not for the small intestine. Remember this. in certain animal demonstrations of there that the 5-hydroxytryptamine or serotonin or somatostatin also actually initiates phase 2 and phase 3 contractions in the duodenum. That is happening in certain animal experiments. We have got that result. Okay, so that was about motility and serotonin. Now coming on to the movements in small and large gut. What are the movements? We see small intestine, there will be mixing or segmentation contraction movement. These will be localized concentric contractions that are happening at certain intervals. Okay, because food is coming, so slowly there will be this mixing movement, slightly it will happen, segmentic contraction will happen, and food will eventually pass from one segment of intestine to the other segment of intestine. There is of course propulsive movement, okay, of the small intestine that will propel the chyme forward. Then is the movement caused by the muscularis mucosae. This is important because muscularis mucosae will constantly, this actually the villi are there which are present, so that villi will actually contract, and that will also help to push the intestinal contents. Okay, and this villi movement will cause contractions of the villi by shortening, elongation, and shortening helps the villi to promote lymph fluid, because remember that the fat, they are actually going into the lymph. Okay, fat is absorbed into the lymph via lacteals, so that villi movement is very important, that is happening. Now talking about the large intestinal movements of the colon, you know that large intestine has certain features. From the external features of large intestine only you can understand that there is presence of gastrations, then there is presence of tinea coli. Okay, there is presence of gastrations, so this will actually cause mixing movements in the colon, and of course there will be involvement of the circularis and the longitudinal muscles because they are throughout the entire length of the GIT, and these bag-like pouches, they are called as the gastrations. Now propulsive movement is the mass movement that happens. You can easily imagine this thing that there should be continuous propulsion, so that is constantly happening throughout the GIT, starting from the esophagus, then in the portions of stomach, then in the small intestine and large intestine. So there will be this peristalsis, a propulsive movement always present, and that will move, and this will propel ultimately the fecal matter. Now this fecal matter, here I am just discussing in short about the defecation reflex that can be directly asked, so always try to write your answers in this tabular format or in this flowchart format so that you can get more marks. Now see over here, defecation. So after food has been digested, absorption is happening, the unwanted products, they are moving down from the ileum, they are moving into the colon. So there is the ileocecal valve present, first into the cecum, then into the colon, then into the rectum. In the rectum, basically the anus, before the anus, the rectum. In the rectum there is only presence of feces. There is further water absorption from the rectum that is happening, so that the feces become actually coherent and solidify to some extent. Okay, so that is very important. Now talking about if the feces enter the rectum, there will be distension of the rectum wall, so afferent signals will actually pass through the minatory plexuses, and that will initiate further peristalsis. That is the descending portion of the colon, the sigmoid portion of the colon, and the rectum. Colon has again four segments are there. One is the ascending colon, then we have the transverse colon, then we have the descending colon, then we have the sigmoid colon. So these four parts of the colon, they are present, and into the rectum, and this will force the feces more towards the anus, because whatever is remaining in the large gut there, that is the fecal matter. Water reabsorption happens from the large gut to a large extent, so that feces actually solidifies to a certain extent. So that peristaltic waves will approach the anus, and there will be opening of the internal anal sphincter. That will relax, because this internal anal sphincter, that is always involuntary, that is not under our control. External anal sphincter is in our control. External anal sphincter, if it is voluntarily relaxed, then there will be defecation. Now the parasympathetic defecation reflex, that is this parasympathetic stimulation, that is very important. Like the nerve endings of the rectum, they will be stimulated, signals will be transmitted by the spinal cord, that is by the pelvic nerves. They are going to the descending colon, sigmoid colon, and the rectum. These parasympathetic nerves will again further travel back to the pelvic nerves and intensify the peristalsis action and relax the internal anal sphincter. That will be completely under, remember this parasympathetic control. Defecation is basically parasympathetic. Okay, remember this. And ultimately, if there is voluntary relaxation of the external anal sphincter, so defecation will occur. So remember these things, these are important. Okay, this reflex can be asked in your exam. So defecation reflex, you should know. You should also read about small movements of in the small and the large gut. This migrating motor complex, this is asked in the Viva exam, and also it is important for the theory paper. Importance of gastric emptying, this is important. Okay, so questions can be asked from these segments. And what is this basic electrical rhythm? This is also important. BER is important over here. Okay, so with this we end the discussion on gastric motility. Okay, gastrointestinal motility, we are ending the discussion. Thank you. Saptarshi Biswas from Murshidabad Medical College, and today we will be discussing about the exocrine pancreas. We had discussed about the associated glands of the digestive system, where we talked about the salivary glands. Next we are discussing about exocrine pancreas, and then we have liver. Now under exocrine pancreas, please remember that pancreas is basically a mixed gland, or it is also an exoendocrine gland. So the exocrine part, that is related with the acinar cells producing the digestive juices of the pancreatic enzymes, whereas the endocrine part, that constitutes less than 1% of cells scattered in the pancreas, that is basically the islets of Langerhans, and they have the respective cells like alpha cells, beta cells, delta cells, and they produce the respective hormones. Now talking about the exocrine part is mainly responsible for production of pancreatic juice from pancreatic acini. If we look into this diagram, we will find that these structures present over here, these are basically the pancreatic acinar cells. Okay, these are the pancreatic acinar cells present in most number, and they will produce these enzymes, and along with bicarbonate. Okay, so bicarbonate will also be produced, and this bicarbonate and all will come into this particular, this is called as the main pancreatic duct. Okay, this is the main or the long pancreatic duct, and from this actually opens into this region. This is actually a part of duodenum. Now see, this is the ending of the stomach region. This almost ends at the level of pylorus, and then we have the first part of duodenum. Then this part is actually the second part of duodenum, which actually houses the pancreas, and this part has this region. This region is actually called as the major duodenal papilla. Okay, this region is called as the major duodenal papilla, and actually it opens through this region. This part is actually called as the ampulla of Vater, okay, or the papilla of Vater. This dilated portion, this part is actually called as the ampulla of Vater or the papilla of Vater. And over here it is surrounded by sphincter, and this particular surrounding sphincter over here, this sphincter is called as, this surrounding sphincter region, this is actually called as sphincter of Oddi. So actually over here, if you see, this is the common bile duct. Okay, this is the common bile duct, and from here bile is also coming and draining into this region, and from there both bile and the pancreatic juices, they are released into this region of ampulla of Vater, into the second part, that is the major duodenal papilla. Okay, now secretin, here it's in the diagram, it says the secretin causes copious secretion of pancreatic fluid and bicarbonate cholecystokinin. This we will discuss later. Now talking about the composition of the pancreatic juice, whenever we are talking about a particular secretion, maximum in the body there is water, and then there are certain solutes also. There can be the inorganic solutes as well as the organic solutes. Under inorganic solutes we have mainly three ions: sodium, potassium is less, very less, mainly sodium, bicarbonate and chloride. Now maximum amount is mainly of the bicarbonate. So if they ask you what is the pH, approximate pH, then you say that it is mainly alkaline. Pancreatic juice is mainly alkaline due to the presence of large amount of bicarbonate. The concentration of bicarbonate in this juice is approximately 145 milli equivalent per liter. Okay, remember this. This is an approximate value. And other than that, there are proteolytic enzymes also. See, on the organic parts we have this proteolytic enzymes, we have lipolytic enzymes, we have amylolytic enzymes and trypsin inhibitors. And mucin is always there. Mucus production happens, and there is a mucin protein present inside it. Now talking about the different proteolytic enzymes. So here the proteolytic enzymes, okay, the proteolytic enzymes over here, these are basically trypsin, chymotrypsin, then we have carboxypolypeptidase or carboxypeptidase. Okay, then under lipolytic enzymes we mainly have, under lipolytic, let's write over here, it's lipolytic enzymes, we mainly have pancreatic lipase. See, one thing was there, gastric lipase, but see, pancreatic lipase is important because after emulsification only, then the long chain fatty acids will actually be broken into further units for better absorbance into the lymph. Okay, so that's why remember the pancreatic lipase. Gastric lipase was there, but gastric lipase was insufficient to digest and break down the larger fat molecules. Then we have cholesterol esterase, and we have phospholipase. So these are the various lipolytic enzymes present. And then under amylolytic, we have the pancreatic amylase. So here we have the pancreatic amylase, okay, amylolytic. And now talking about the trypsin inhibitors. Now see, whenever the first synthesis in the pancreatic cells, if there is trypsinogen which is produced, okay, then chymotrypsinogen, pro-carboxypeptidase, these are basically the zymogens, or that is the inactivated enzymes that are produced, and they will only be activated when they are secreted into the intestinal tract, that is they are reaching the duodenum, and they are actually activated by the enzyme enterokinase. Remember this, that all this, they are actually mainly activated by a particular enzyme, that is enterokinase, and enterokinase is released from the intestinal mucosa. It is released from this intestinal mucosa. When is it released? When actually chyme comes in contact. Now what is this chyme? That was the thick acidic paste. Food bolus was coming into the stomach, and after churning and all processes in the stomach, now the thick acidic paste is released slowly at certain intervals into the duodenum, and that is happening in the form of chyme, which is a thick acidic paste. And this will happen, this enterokinase will actually release from the intestinal mucosa when chyme is coming in contact with this mucosa. When chyme is in contact with mucosa. Okay, so trypsinogen also can be autocatalytically activated by trypsin also, that was already formed over there, that is from trypsinogen into trypsin, which was already there, so that can also further autocatallate itself. Okay, and activation can happen. Chymotrypsinogen is later produced from trypsin, and similarly carboxypeptidase converts into carboxypeptidase. Okay, now see, secretion of trypsin inhibitor. One other important thing here I have written, that was trypsin inhibitor. Now what is this trypsin inhibitor? Let's explain over here. Okay, now what is this trypsin inhibitor? Now see, whenever these pancreatic enzymes are produced, remember this particular organ from where it's produced, the gland, that is basically, that is also having different tissues which can be digested. So it's important that the proteolytic enzymes of the pancreatic juice, they do not activate over there in the pancreas itself and lead to the destruction and damage or digestion of the pancreas. Okay, so that's why we basically have this trypsin inhibitor that is released from the pancreas, and this is actually found in the cytoplasm of the glandular cells and prevents activation of trypsin. So what does it do? This prevents activation of trypsin. Remember this, this prevents activation of trypsin, and this is very important to prevent auto digestion of pancreas, because pancreas could have been digested by its own secretion. So that's why this is done, this is important. and this trypsin that activates later, this trypsin will activate. If this trypsin is inhibited, then other pancreatic enzymes will not be activated, because trypsin actually activates others. And one of the essential factors for trypsin activation is enterokinase or enteropeptidase. Okay, so when the pancreas becomes severely damaged, or when the duct becomes actually blocked, so large quantity of the pancreatic secretions become remain accumulated in the damaged regions. So this effect of the pancreas inhibitor is often overwhelmed, and in this case the pancreatic secretions rapidly become activated and literally digest the entire pancreas. So this condition is called as acute pancreatitis. This is actually a medical emergency. What can happen? See, blockage can happen. Now when we will discuss later, we can discuss about the pathology also, when we will read in second year about pathology. That time we talk about these type of pathologies, that anywhere there can be malignancy, anywhere there can be occlusion or obstruction, anywhere there can be other genetic reasons also for changes of the particular cells. So if that happens at the level of this pancreas, so what will happen? There will be acute pancreatitis. So in this case, what happens in acute pancreatitis? What happens? The activity of trypsin inhibitor is lost. So trypsin inhibitor activity is lost. Activity is lost. Why is it lost? Because see, when there is already excess amount of pancreatic juice present, so that particular inhibitor is not sufficient to actually inhibit trypsin to that level. Trypsin inhibitor at that point of time becomes inefficient. It cannot inhibit the entire amount of trypsin that is produced, because trypsin is not released into the pancreas and into the duodenum, because already there is a blockage or any type of other pathology. So that's why what will happen? Inflammation will happen. This so trypsin will be there. So trypsin activates other proteolytic enzymes. So it will activate the other proteolytic enzymes, and what will happen? There will be damage. Itis means inflammation. So there will be damage to pancreas. Okay. So this can happen in acute pancreatitis. So this is a medical emergency, and it is at times very lethal also. Now talking about the production of this bicarbonate, as we are discussing about this bicarbonate and how is it important and how is it coming into the pancreatic lumen, let's see and let's discuss it. Now see, in this capillary interstitium, so capillary beds, these have larger amount of carbon dioxide. So carbon dioxide diffuse like this, and then this carbon dioxide will enter into the pancreatic cell. Carbon dioxide crosses the lipid membrane, phospholipid membrane, and enters. Now there is already water present inside the cell, so it will combine with water in the presence of carbonic anhydrase enzyme. This will form carbonic acid. Now carbonic acid will further dissociate into this. Okay, now see what will happen. This is the first step. Now see, there will be exchange of this bicarbonate. This is produced. So this bicarbonate will actually exchange for chloride. So chloride will come in into the pancreatic lumen, bicarbonate will go out. So in this case, here there is presence of one particular protein that is basically called as, this particular protein is actually called as bicarbonate chloride exchanger. This is an anion exchanger. Okay, so here this exchange will happen mainly on the luminal surface. This thing will happen. Now see what happens to this H plus. Now H plus is there. So H plus, now in this case it will get exchanged for sodium. So here there is a cation exchanger. So this is a cation exchanger. So here sodium will enter the cell, and here hydrogen will lost into the interstitium. Okay, so is this much clear? Sodium will be coming into the cell, pancreatic cell, whereas H plus will be lost over here into the interstitium. Okay, and so here what is present? There is basically another transporter present, that is sodium hydrogen antipode. This is called a sodium hydrogen antipode. Now see over here, sodium is accumulating. So sodium will accumulate. So automatically what will happen? There will be activation of another pump. So this sodium will be sent out, and in this case potassium will be taken in, and this requires a pump. So this is the activity of sodium potassium ATPase pump. So this neutrality and all this has to be maintained inside the cell. So when sodium is accumulating, sodium will now move out. It will be forcibly thrown out of the cell against the concentration gradient active transport, which requires ATP. So ATPase will be actually, ATP will be used. ATPase actually will break ATP, hydrolyze ATP, and that energy will be required to transport sodium from pancreatic cell into the interstitium, and in that exchange for potassium. Okay, so is this much clear? Now talking about what was happening, I have discussed about sodium hydrogen exchanger. Also remember, there is a co-transport that is happening. So whenever some amount of sodium is entering, bicarbonate will also freely diffuse. Bicarbonate which was present in this interstitium, this will also freely diffuse into the cell, and in this exchange what will happen? Sodium will also be getting entry. So here there is presence of a sodium bicarbonate co-transport. Okay, that is entering into the cell. Now here I have discussed on the luminal side, this was the chloride bicarbonate exchanger, and sodium water, they are always freely transporting. So sodium will also move into the pancreatic lumen. So here sodium also moves from this side into this side, and in this case sodium also brings it with water. So water is osmotically active particle, sodium will bring water along with it. So overall what is happening, the negative voltage of the lumen also pulls the positively charged sodium across the tight junctions between the cells. So this is what, this is movement through tight junctions. Okay, so this is the movement through tight junctions. Overall movement of sodium and bicarbonate from the blood into the ductal lumen, that actually creates an osmotic pressure gradient, and water is actually dragged by the osmotically active particles. And like this, there is almost completely isoosmotic bicarbonate solution. Mainly this side has, the pancreatic lumen side has bicarbonate that is isoosmotically present. Okay, now talking about the regulation of the pancreatic secretion. First, the stimuli. Now see, the basic three stimuli that are important for the secretion. First comes from acetylcholine. Okay, this comes from acetylcholine. Now from the parasympathetic vagus nerve endings, this is released. Then cholecystokinin, pancreatinine, that is actually secreted by the duodenal and the upper jejunal mucosa when the food is entering the small intestine. Okay. Then we have the secretin. So secretin is secreted from the duodenal and the jejunal mucosa, and it's happening when the highly acidic food is entering into the small intestine. The first 2 to 3 stimuli, acetylcholine, CCK, and these stimulate the acinar cells of pancreas, causing production of large quantities of pancreatic digestive enzymes, but relatively small quantities of water and electrolytes also to go with this enzyme. Without water, most of the enzymes actually remain temporarily stored in the acini and ducts until the fluid secretion comes along to wash them into the duodenum. Secretin, in contrast, remember that is secretin in contrast to the first 2 basic stimuli, stimulates the secretion of large quantities of water, of sodium bicarbonate by pancreatic ductal epithelium. So that was what written. Now talking about the multiplicative effects of different stimuli. So when there are these 3 different stimuli of the pancreatic secretion occurring at once, the total secretion will be of course far greater than the sum of the individual secretions. Separately they are secreting, and when they are all 3 as accumulated or associatedly, they are producing an action, so that will of course be greater. So this much should be clear. Now discussing about the phases of pancreatic secretion, first is cephalic phase, then we have the gastric phase, and then we have the intestinal phase. So in the cephalic phase, remember this is during this phase of pancreatic secretion, it is the same nervous signals. So this is basically the same nervous signal from the brain that cause secretion in the stomach. That cause secretion in the stomach. Okay. And also causes acetylcholine and released by the vagal nerve endings of the pancreas. So causes vagal release of ACH. Okay, so there is vagal release of ACH. This signaling causes moderate amount of enzymes to be secreted. So in what will this cause? This will cause moderate amount of enzymes to be secreted. Approximately, that is about 20%. Okay, that is approximately 20%. However, little secretion also flows immediately to the pancreatic duct into the intestine, because only small amount of water and electrolytes are secreted along with the enzyme. Now talking about the gastric phase, during this phase the nervous stimulation of pancreas continues. Okay, accounting for another 5 to 10%. So gastric phase there will be next 5 to 10% production. Next 5 to 10% secretion that will happen. Okay, accounting for another 5 to 10% after meal. Then in the intestinal phase, after chyme leaves the stomach and enters the small intestine, the pancreatic secretion becomes copious. Okay, mainly because of secretin. So in the intestinal phase, there is presence of secretin. I have told you secretin is very important. So in secretin's presence, what will happen? There will be copious secretion of this juice, of pancreatic juice. Remember, this secretin also causes large quantity of pancreatic juice with bicarbonate. Okay, higher quantities of bicarbonate that will be coming out with it, and low concentration of chloride. So higher concentration of bicarbonate and low concentration of chloride. Okay, the carbonic acid then immediately dissociates into CO2, and this CO2 is back absorbed into the blood. What actually happens, the HCl which was present, the net reaction, and in this case sodium bicarbonate, as I've told you over here, that sodium and bicarbonate both are coming. Okay, so sodium and bicarbonate in the pancreatic juice. So when they're coming, this reaction is happening, and over here there is formation of NaCl and H2CO3. Now carbonic acid, this will actually split up into carbon dioxide and water, and this carbon dioxide will flow into capillary. Okay, this will go back to the capillary. So this one should be clear. Now cholecystokinin, that is the role of hormones. Under that we talk about that cholecystokinin also contributes to the control of the digestive enzyme secreted by the pancreas. So cholecystokinin, now let's discuss about it. How does this cholecystokinin contributes? So over here, see, cholecystokinin will contribute to the control. So when there were food in the upper small intestine, another hormone, that is cholecystokinin, which is released from a group of cells. So eye cells, from the eye cells there is release of CCK. Okay, this release of CCK results especially from the presence of proteases and peptones. This happens due to presence of proteases and peptones, which are basically products of the partially digested proteins, and also certain long chain fatty acids LCFAs. Okay, so cholecystokinin like secretin, they also pass way of the blood pancreas, but instead of causing sodium bicarbonate secretion, it mainly causes secretion of much more pancreatic digestive enzymes by the acinar cells. So remember that when we are talking about secretin was responsible for higher concentration of bicarbonate in pancreatic juice. Okay, it was responsible for production of higher quantity of bicarbonate. But remember, eye cells that release CCK, that mainly this causes excess production. This causes excess production of pancreatic juice. Okay, so this difference between pancreatic stimulatory effects of secretin and CCK, this should be very clear that how they are different. So this is important. Okay, now talking about this picture which shows that how these things are happening. So here see, amylase is working on this starch, and that is breaking down this starch into finer particles, like that is the dextrin and maltase. Okay, maltose into dextrin and maltose, then maltose under presence of maltase, that is further breaking down. So these discussions I will do later when I will discuss about digestion and absorption of carbohydrate, proteins and fats. Till now just remember the roles of this hormone secretin that I've discussed. CCK, PZ released from the eye cells. These are very short topics. Just have a gist idea that what is overall thing and just go for it. Okay, remember this bunch. This is important. And how there is production. This should be clear. The concept is more or less same that is happening. That mainly the sodium potassium ATPase pump is present on the basolateral side, whereas this exchanges, anion exchanges and all, they're mainly present on the luminal aspect. So this basic concept should be clear so that in renal physiology, digestive physiology, wherever it comes, you actually understand the basic concept that how these productions and secretions are happening. So this part is very important and this should be very clear. Now talking about these things, these are actually asked in your Viva exams more, and you should be familiar with this concept, that is how this thing is happening. In slow amount of this pyloric sphincter is opening and small amount of food, that is basically the chyme, that is released into the first part of duodenum, that is happening. So this is very important, and always remember that when you are discussing about a particular secretion, always remember about the nervous control. That is very important. So with this we will sum up the pancreas, exocrine pancreas and its function. Now later when we will discuss about how different proteins are broken down by these proteolytic enzymes, that we will do later. Okay, we have a dedicated chapter for that, a dedicated lecture for that. Thank you. Everyone, I am Saptarshi Biswas from Murshidabad Medical College. Today we will be discussing about the various functions of liver. Now liver is the largest gland of the human body, we all know, and it produces many substances and it has a key role in metabolism. Now talking about the microstructure of liver, we all know from anatomy. Okay, I am not going into the details, but this is the structure of a hepatic globule, which is in one of the classification it is said to be somewhat hexagonal in shape. Over here in the center we find a central vein, and over here these are the radiating hepatocytes. This is basically the bile canaliculi, and surrounding this we actually have the hepatocytes. Okay, now at these corners or at these ends we basically find the portal tract, which includes the hepatic artery branch, we have the portal vein branch, and also we have the bile duct. Okay, all these bile ducts from various hepatic globules, they will fuse and they will form the hepatic duct. Okay, the right and the left hepatic duct, they will fuse and form the common hepatic duct. Now another duct coming from the gallbladder, that is called as the cystic duct, that will actually join with this duct and form the common bile duct, and later they will drain. Now this probably we are all familiar with, so I'm not drawing over here and not going into the details. Now talking about the liver functioning as a blood reservoir. Now so liver is an expandable organ. Okay, and also remember that liver has the capacity to regenerate. Okay, here if you say that why is it an expandable organ, because a large quantity of blood can be stored inside the blood vessels. Over here if we have these vessels, we have the hepatic sinusoids, so in these regions huge quantities of blood can be stored. 10% of the body's total blood volume, that is approximately 450 ml, is present in the hepatic veins and the sinuses. Okay, in high pressure condition of the right atrium, there is a back pressure that happens in the liver, because what happens, if you have seen the diagram of the liver, you must be familiar with that. If this is supposed the liver, then from here is the emerging inferior vena cava. So inferior vena cava, it will join with the superior vena cava, it forms the vena cava trunk, and that will drain into the right atrium of the heart. So what happens, if high pressure occurs in the right atrium, then automatically at that point of time blood will not go into the liver, that is not go into the heart, so there will be back pressure that will develop in the liver, and there it can approximately accumulate 0.5 to 1 liter of that extra blood. Okay, so this is important that it acts as a blood reservoir. Now talking about the macrophage system, you see liver has specific macrophage cells. These are called as the Kupffer cells. Okay, these cells are important, like each and every region, almost every region have the specific macrophages, and for our convenience we have named them accordingly. Like the macrophages in the livers, they are called liver cells. Surrounding the liver cells, they are called as the Kupffer cells. Okay, they are the large phagocytic macrophages. So what are Kupffer cells? Kupffer cells are large phagocytic macrophage. Okay, and they line mainly in the hepatic venous sinuses. That line in hepatic venous sinuses. Okay, so what actually happens, this is required because whenever we are taking in food, from our basic knowledge we know that the food that is passing down into the stomach, and from the stomach it is going into the intestine, and remember in the intestine there is absorption of food. So absorption not of the food, that is of the nutrients, they are broken down, that is the glucose, the fructose, then amino acids, proteins, they will all fats, they are all going into the circulation, intestinal circulation, and from there they are going actually into the portal circulation. Via the portal circulation they are going to the liver. Now they can actually carry bacteria, so there is requirement of actually killing the bacteria. There is requirement of activation of our immune system via activation of the macrophages, that is the Kupffer cells. Okay, because what will happen, if these bacteria they go into the small intestine, they are already present. If they go into the large intestine, what will happen, there will be interference of the already present colon bacteria and the bacteria that is entering with the food. Though there are other barriers also which are preventing the bacterial entry, but certain bacterial infestation can happen easily, and so that's why that is a type of defense mechanism, that yes, if the food is getting absorbed, so automatically the bacteria, they will also, they are much smaller, they will also enter into the enterocytes. Through the enterocytes they will go into the portal circulation, so they must be destroyed at the level of the liver. Okay, so this is the diagram given here. From this diagram I was talking about the microstructure of the liver. There you find over here the hepatic artery, we have the portal vein, the bile duct, all these have connections present, and here we see the spaces of DC, spaces of mole, these are all present over here. I'm not going into the details, but just for an overall idea. Now talking about the metabolic functions of the liver, we all know that it is a key function in carbohydrate, protein and fat metabolism. How the entire metabolism is integrated, I'm not going into the details, but remember the processes, that what processes are happening. That is, first there is gluconeogenesis. For glucose there is gluconeogenesis. Okay, new synthesis of glucose from other products like from glycogen. Then there is glycogenesis. Okay, in the liver, in certain muscle cells there is glycogenesis. Here glycogen is produced from the glucose and that is stored. It is important. Then there is actually fatty acid entire metabolism that is happening. Fats are broken down, triglycerides, fatty acids, lipoproteins, all these things are happening, and also protein metabolism. Certain proteins, certain enzymes, they are specifically present in the liver. Okay, the liver is the seat of the urea cycle because certain enzymes they are present in the liver and they will be required over there. That means the circulation is happening, through that the substances are coming, and in this there is formation of the urea. Okay, that is a metabolic waste and that has to be removed from the body. Liver has large stores of ferritin. Okay, liver stores ferritin. Okay, when after heme actually that is the highest amount of iron binding, anything that is bound to the iron, that is first is the heme, then we have the ferritin. Okay, remember like this. Hepatocytes, they have a large amount of protein, that is the apoferritin, that is capable of reversibly combining with iron. Now this actually talks about a rough picture of the iron metabolism that we will be learning in biochemistry, but that is quite important. Okay, because that is related with several kinds of anemia, several disorders, then their medications, all these things are integrated. Liver also forms major proteins in blood coagulation factors, then certain plasma proteins. Okay, they are all produced by liver. So over here fibrinogen, prothrombin, accelerator, globulin, factor 7, these are produced. Okay, then other than that, certain blood proteins, like if you say about albumin, plasma proteins, the plasma proteins like albumin, globulin, their production also happens in the liver. Albumin, globulin production. Okay. Now drug metabolism, there is drug interference, okay, interaction of different drugs, and also certain enzymes are there, liver based enzymes are there, the CYPs enzymes are there, they are all dealt in drug metabolism. I'm not going into the details. Talking about the bile secretion of the liver, now see this is mainly theoretical topic, and here question can be directly asked in your Viva table that what is the normal amount of bile secreted by the liver or synthesized by the liver. You will say approximately 600 to 1000 ml per day. Okay, because it is important. See, entire amount of bile that is produced in the liver, that is not directly used because that is concentrated in the gallbladder. Gallbladder, it is stored and it is concentrated over there, and after that amount only the bile is released slowly. When there is requirement, when there is fat present in the intestine, then only there will be requirement of this bile. So at that point of time bile will be released. Okay, bile salts. Now these bile salts, there are the bile acids, glycolic acid, taurocholic acid, and their salts reacting with sodium and potassium, we get the sodium potassium glycolate and taurocholate. These are very important because these have key role in emulsification of the fats. Now what is emulsification? Actually the larger fat molecules, they are broken down into droplets. If this was a fat molecule, this has to be broken down into finer droplets, so that what will happen, the lipases will act on them in the intestine. The lipases can act on these and they can actually break down this fat into certain fatty acids and triglycerides. Okay, fatty acids and triglycerides. So this is important. After that only there will be presence of cholesterol esterase, they will act, there will be phospholipases. All these are dealt in the fat metabolism in biochemistry. Okay, I'm not going into those details, but remember emulsification is mainly the process of breaking down of the larger fat droplets into finer or smaller droplets, such that the lipase is released, that is the intestinal lipase that can act on these and break down the fats. Okay, so this is the structure. Here the liver is present. Now see, from the stomach, this is basically if I mark over here, this is basically the common bile duct. Okay, this is the CBD. It is coming and it is entering over here. They all actually drain into this region of the major duodenal papilla. Okay, this region is called as the major duodenal papilla. Okay, this segment is called as the major duodenal papilla. Now over here, this actually fuses with this, and here there is actually formation of the hepatopancreatic duct. See, this is the long pancreatic duct that is coming. Okay, this is the long pancreatic duct that is coming. This is the common bile duct. They will fuse together and they will form the hepatopancreatic duct. They will form the hepatopancreatic duct. But remember, every time there is not presence of bile in this CBD, because over here there is actually a valve at this point that is actually called as the sphincter of Boyden. Okay, remember at this level we actually have something that is called as the sphincter of Boyden. Last, when we were discussing about the pancreatic secretion, at that point of time we talked about there is presence of this ampulla of Vater in this region, and that is surrounded by the sphincter of Oddi. Okay, but here we have the sphincter of Boyden present, such that the bile that actually is coming down via this route, it is coming down via this route. Okay, so that it is actually remaining over here, and there is backflow of that bile into the gallbladder, and over here the bile remains there, it is concentrated at that level, and when there is requirement, that time the sphincter of Boyden will relax, and that will allow slow amount of bile release into this hepatopancreatic duct, and ultimately it will drain into the major duodenal papilla. Okay, so this should be clear. See, bile stored and concentrated up to 15 times. Concentration is very important. That amount of bile that was released from the liver, that was about 600 to 1000 ml, but that is not the entire amount, because that contains higher amount of water, but that has to be reduced, okay, such that the fats can be emulsified, because you know that fats and water, then of course fats are hydrophobic in nature. Bile acids via the blood is stimulating the parenchymal secretion also. Vagal stimulation causes weak contraction of the gallbladder. Now I will explain it later. I'm not going into the details now. Secreted by the bloodstream stimulates liver ductal secretion. Secretin is very important because secretin actually stops the release of the pyloric sphincter, and it rather allows the opening of the sphincter of Oddi, such that the secretions from the pancreas, they can move, because some amount of that is chyme has come into the duodenum. Okay, so it has to prevent the remaining portion to enter. That's why it will now close the pyloric sphincter for some time, and it will allow the opening of the sphincter of Oddi, such that here there is release of both bile and pancreatic juice, such that the pancreatic juice acts on the chyme. Okay, this is important, and that is stimulated by the local hormone secretin. Talking about this flow chart, see the hepatocytes, they are producing the bile acids, glycolic acid and taurocholic acid. Okay, they are coming to the bile canaliculi. If you remember the bile canaliculi, the hexagonal hepatic lobule, it had the central vein, and these channels which were present, these channels are actually the bile canaliculi. Okay, they have the surrounding hepatocytes are surrounding these regions where actually the bile production is happening, and they're draining into the bile canaliculi, and from there, this bile canaliculi, they're entirely draining over here into the bile duct, because the portal tract included the bile duct, the hepatic artery, and the portal vein. Okay, so here in this, the bile duct they're coming, and after that all these bile ducts, they're forming the terminal bile ducts, and they're draining into the hepatic duct. From there, as I've told you, the cystic duct from the gallbladder that is coming and forming the common bile duct, and after that the common bile duct, so over here it is coming, so the pancreatic duct that was coming, the long pancreatic duct that was joining with this and forming the hepatopancreatic duct, which was ultimately draining into the ampulla, major duodenal papilla in the region of ampulla of Vater via the sphincter of Oddi. So this part of anatomy must be very clear. Okay. One thing, remember that the maximum amount of volume of bile that the gallbladder can store is basically the 30 to 60 ml. Gallbladder is present basically in the fundic lobe, that is the fundus of the gallbladder that is lodged into the liver portion on the undersurface of the liver. There we will find the gallbladder present, and there is the presence of the callout strangulum and all. I'm not going into those details. They're surgically important when you actually have to laparoscopically remove the gallbladder, that is cholecystectomy is done. So at that point of time, that callout strangulum and all those regions and the undersurface of the liver, they are very crucial. Okay. We're talking about the composition of the bile. So here you see the maximum amount of water is present again. Any secretion from the body has maximum amount of water only present. Okay, and you see that it is concentrated at this level such that now the water's amount becomes 92 gram per deciliter when it is coming in the gallbladder. Okay, then this is actually not important because they will not ask you. But remember the composition in terms of these things like the bile salts. What are the bile salts? They can ask. Okay, in the exam, that what are the bile salts present? Means in the viva table, the teacher can ask you, the professor can ask you that what are the bile salts present. You will have to name them: sodium and potassium, glycolate and taurocolate. Then there are presence of bilirubin. Now, which form of bilirubin is actually present in the liver? There is the conjugation of the bilirubin is happening. Okay, the conjugated or direct bilirubin, that is only mainly coming in the common bile duct and it is stored in the gallbladder. So remember that mainly it is in the form of direct bilirubin. Cholesterol is present because cholesterol is important. When we see gallstones, what are these gallstones? They are cholecystolithiasis. These are basically the condensations and that is the reactions are happening, and this cholesterol, excess amount of cholesterol, they are converted into stones. And these stones, they are actually present. If they are present over here, it's okay to some extent. But if these small stones, they are actually coming and blocking this region. Okay, if they are coming and blocking this opening of the cystic duct, or there it is a big particle and it is dislodging over here. So what will happen? There will be the bile that is directly coming, or it can flow into the common bile duct at this level, and there can be obstruction at this level. So this is very sensitive because when bile will not flow, so automatically fats will not be digested. So there are chances of fats present in the stool. So that condition is at times that can happen, and there is severe pain also at times because this obstruction is happening. So that is important. Gallbladder stones, that has surgical importance also. Now talking about certain other substances like lecithin that is present. Okay, then certain ions. So these common ions are always present. Talking about the role of CCK, cholecystokinin. Okay, in gallbladder emptying. So what happens? There is a rhythmic contraction that is happening every time the gallbladder wall, and that leads to the relaxation of the sphincter of Oddi at the region of the ampulla of Vater. So cholecystokinin will actually stimulate the secretion of the digestive enzyme to acinar cells of pancreas that we have already discussed, and that will also stimulate the gallbladder to release bile. So remember, cholecystokinin, secretin, they have almost similar functions, that they will actually prevent gastric secretion. That means they will not allow the gastric contents to enter the intestine, but whatever is present in the intestine, they will first try to digest that. That is, they will stimulate the pancreas to release the pancreatic juice, the intestine to release the intestinal juice, so that there is actually first digestion of the food, entire fat, protein, nucleic acids, then also we have the carbohydrates, entirely they will be digested, and then so this process should continue so that there is not excess amount of crowding happening. Okay, so that is important. Now talking about the function of bile in fat digestion and absorption, this is important, that how bile salts are actually regulating fat digestion and absorption. So I will discuss this now. Now see, liver cells, they produce approximately 6 gram bile salts daily. Okay, now how is this happening? The precursor of bile salt is coming from cholesterol. Okay, this is coming from cholesterol, that is either coming from the diet, or cholesterol is endogenously synthesized in the body. So cholesterol, that is first converted into cholic acid. Okay, this cholic acid, or it is also called as chenodeoxycholic acid. Okay, so cholesterol is first converted. So these convert actually this turn combined principally with the glycine and with lesser extent to taurine. So there is of amino acids glycine, mainly glycine, okay, and also to some extent with the amino acid taurine. So when this is happening, so they will actually form glyco and tauro conjugated bile acids. So there will be formation of glycine and taurine. So there will be glyco and tauro conjugated bile acid formation. Okay, so then what will happen? They will react mainly with sodium, and this portion they will react with the sodium or potassium, and they will form what? Sodium glycolate, or there can be sodium or potassium glycolate and sodium and potassium taurocolate. Mainly glycolate salt is there. So this is how there is production of okay, the bile salts from cholesterol. So see, these questions, they can be asked in your viva table. At times very simple MCQs are asked, that from where are you getting the bile acids? If you're talking about the bile salts, then from where the bile acids are coming? So cholesterol. It's a very simple answer, but you should know these facts. Okay, now see what will happen. They have a detergent action on the fat particles in the food, which decreases the surface tension of the particles and allows agitation in the intestinal tract to break the fat globules into minute sizes. This is actually emulsification process. So what was the function That was emulsification. Now what is actually happening? They have always write that bile salts have detergent action. So bile salts have detergent action on fat particles. And how is this happening? Whenever this will actually lead to decrease in surface tension, there will be decrease in surface tension of fat particles, and this will allow agitation in intestine. Okay, so that was about emulsification that allows agitation in the intestinal tract. Okay, now talking about the enterohepatic circulation of bile salts. Enterohepatic circulation of bile salts. See over here what is happening. So about 95% of the bile salts, they are reabsorbed into the blood from the small intestine. Okay, and about 1/2 it is through the diffusion through the mucosa, that is in the early regions of the small intestine, and remaining that happens by the process of active transport through the intestinal mucosa in the distal part of the ileum. So here you see I have discussed over here 3 routes. I have just mentioned 3 routes. Mainly there was this if I write over here, enterohepatic circulation. So first point is 94% is reabsorbed into blood from the small intestine. Okay, that is happening, and that is about 1/2 of it is happening through the mucosa in the early portions of the small intestine by diffusion from early portions of small intestine. Then we have about some amount of it is actually activated by the transport, that is active transport is happening over here, and they are entering active transport through intestinal mucosa. Okay, and remaining from the ileum. Okay, from the ileum there is again reabsorption. So I've discussed 3 routes. So mainly what is happening, if you look at this diagram, you will find this bile acids, they reabsorb from the intestine. Okay, so these bile acids, they are coming, this is the primary bile acids, and they are again reacting over here, the bicarbonate reserve is there, and from there what is happening, you see from here, if you see that here the conjugation process is happening and it is moving via this region, there is the bicarbonate co-transporter present at this level. So the bile that was coming from the intestine via this portal system, that is reaching to the level of the hepatocyte, and from here the primary bile acid, that is these acids, they are directly going into this bile duct region, and they are again going into the bile duct. Okay, and remaining some amount that is actually taken up by this over here, there is active transport. So whenever active transport, the sodium potassium ATP is found, which will send 3 sodium outside, that is towards the blood, here in this case the portal venous blood, and it will pump 2 potassium for the expense of 1 ATP hydrolysis. Some amount of secondary bile acids, they are also coming via this bicarbonate co-transporter, and for this they are again going into the circuit, that is bile duct. So this is basically the enterohepatic circulation of bile acids and bile salts. Okay, this is very important. Now that's why it is said that about 94% of this is actually getting recirculated, and that is the enterohepatic circulation. If a bile fistula empties the bile salts to the exterior for several days to several weeks, so they cannot be reabsorbed from the ileum. So liver at that point of time increases its production of the bile salts from 6 times to 10 times. Okay, which is actually increasing the rate of bile secretion, and again they will normalize it. Okay, so this process actually continues. Now talking about the role of secretin. So secretin, what is the role of secretin? So secretin, that was stimulating the release of pancreatic juice. Okay, pancreatic juice from the pancreatic acinar cells. So similarly, that will also stimulate gallbladder to release bile, and there will be relaxation of sphincters to allow bile flow. Now what is happening, there will be relaxation of the sphincter, that is sphincter of Oddi, sphincter of Boyden, both will relax such that this will happen. The bicarbonate that is present in turn passes into the small intestine and joins the bicarbonate from the pancreas, such that there is neutralization of the HCl. That is also very important because bicarbonate has to neutralize the acid that is coming with the chyme. Okay, now talking about, when we have discussed this, now let's discuss about the formation of gallstone. So as I was talking about, that gallstones are basically accumulation of cholesterol. Okay, so what is happening over here, cholesterol precipitates in the gallbladder. Cholesterol will precipitate in the gallbladder. Okay, and the amount of cholesterol in the bile is determined partly by the quantity of the fat that the person is consuming. As I've told you, when there is a higher fatty diet, so cholesterol is already increased, so precipitation will increase, and slowly there will be deposition in the gallbladder. Cholesterol is almost completely insoluble in pure water, but the bile salts and lecithin, which are present in the bile, they will physically make cholesterol from ultramicroscopic micelles, okay, in the form of the colloidal solution. So when the bile becomes concentrated in the gallbladder, the bile salts and lecithin also becomes concentrated along with cholesterol, and this gives the cholesterol in solution. So that is happening. There is formation of this micelles where actually there is involvement inside. There is the particle that is hydrophobic, and surrounding that we have hydrophilic particles like there is lecithin, the bile salts. So salts are basically, they are actually hydrophilic. So water will surround it, and there will be formation of a colloidal solution. So there will be a colloidal solution formed. Now see, there is constant accumulation and precipitation of this cholesterol which was there. So what will happen? There will be increase in production of gallstones. So that is important. So whenever we actually measure the amount, so amount of gall, if there is any type of gallbladder stones present, so first we actually perform an LFT. If there is LFT, the lipid profile, we do these tests, so that if there is already extra presence of cholesterol, if we see that, then there is bilirubin levels are elevated. So all these things we have to assimilate, and then we can go on for an USG, or there can be other diagnostic tests also done to detect whether there is any type of biliary obstruction. Okay, so with this we end our discussion on liver and its function. Thank you. Hello everyone, I am Sadarshi Biswas from Murshidabad Medical College, and today we will be discussing about the digestion and absorption of carbohydrates. So among the food items that we are consuming, most amount is basically carbohydrate. Like from the starting from the morning, we are consuming either roti, chapati or bread. Okay, so that is full of carbohydrate. Then in the afternoon we are mainly following rice, or somewhere it is paratha, or somewhere it is bread. So mostly our diet depends on carbohydrate, because carbohydrate is the main source of energy, because from there we derive glucose. Okay, and glucose is the chief energy source, because glucose will actually be utilized in glycolysis, and after that in the Krebs cycle and all, to produce energy. Okay, other than that, our diet also includes fats, proteins, vitamins, minerals. Okay, these are the essential substances that should be present in each and everyone's diet. Now talking about the digestion and absorption of carbohydrates, first see, what do you mean by digestion? When we are talking about digestion, digestion is not a single step. It is actually a complex process. Multiple steps are there, and that we are going to see. First is mixing and lubrication of the food in the GIT, that is uniform homogenization of the food particles. So that basically we call it as we are masticating the food, we are chewing the food, mastication is happening, and there is mixing of the food. So after mastication, there is this mixing and chewing, all these things are happening, so that there is a uniform homogenization of the food inside the buccal cavity. Okay, after that is the process of breakdown of these complex molecules. Now what is present? The carbohydrate that we are taking, mainly we have starch. Okay, this starch is there, but are we directly consuming this starch? No. In most of the cases this should be cooked starch. Okay, this should be cooked starch, because cooking process will make the digestion faster and easier. We cannot consume directly the starch, because that is our body will not be able to digest it properly. So that's why we are dependent on cooked starch. So this cooked starch is basically a complex polymer. Okay, so that has to be broken down into simpler forms, and like they can be broken down into oligomers, dimers and monomers. Mainly what is required and what is absorbed by the intestine, that is basically in the form of monomers. So the process that happens, that is basically we find that all the digestive enzymes, they mainly act by hydrolysis. Mostly all digestive enzymes act by hydrolysis. This is very true. And after that, most GI tract enzymes, they are secreted as inactivated precursors, and they are later activated by the GIT. After that, we have secretion of acid or bicarbonate from the GI tract that will actually ensure optimal digestion of the particles. That optimal pH is required. So the activation is required, activation is done, certain enzymes are activated, and many enzymes are also deactivated at certain pH. Okay, then is secretion, as I've told you, acid and bicarbonate, that is required. Emulsification of fat by the bile salts. This in the last part we had discussed, that how bile salts, they are actually breaking down this fat. There is detergent action of the bile salts for emulsification of the larger fat droplets. And finally we have digestion of the oligomers, that is happening mainly at the level of the intestine, and there is absorption. Okay, so here what is the process? This is absorption at the level of the villi. There are folds that are called as the valves of Kurtring, and these folds are basically surrounded with the mucus. This mucus layer is there, which has basically these projections villi, and we have attached microvilli on them. So this is basically the anatomy of the small intestine, and this is very important for proper absorption of the food, and this is actually providing that surface area which is required. Now talking about what is the digestion of various food by hydrolysis. When we are talking about that, the carbohydrates in the diet, they are basically the polysaccharides. Okay, mainly in the form of polysaccharides, or sometimes they can be the disaccharides also. Like we consume milk, so milk has that sugar, that is lactose, so that is a disaccharide. Or we actually take combinations of monosaccharide, disaccharide, polysaccharides, so there can be a combination. So remember that how these polysaccharides are formed. They are mainly formed by condensation of the monosaccharide or the disaccharide units. In chemistry, we had already studied in the class 11 and 12 that how these reactions are occurring. That is, the condensation is happening. The monomers, different monomer units, they are combining to form the polymers. They are the polysaccharides. Okay, this phenomena means that when an H plus ion has been removed from one of the monosaccharide and a hydroxyl ion has been removed from the next one. Okay, so like this happens. So the two monosaccharides, when combined with each other, and then there is what at the sites of removal, and the H plus and OH minus combine to form water. So when water is released in a reaction, that is basically called as condensation. That happens, and there is production of this polysaccharides. Okay, but when these carbohydrates, they are digested, this is reversed, and the carbohydrates, they are now converted into monosaccharides. First might be at some point they are converted into the oligosaccharides, then to disaccharides, and then to the monosaccharides. Ultimate unit of absorption should be at the level of the simplest form, that is the monosaccharide. Okay, so specific enzymes in the digestive juices of the GI tract, they will return this H plus and OH minus from water of the polysaccharide, and thereby they will separate them. So this process is actually the hydrolysis. Like see, here we have taken two groups. This is one group. Okay, this is one group, alkyl group. So this R double dash and this is single dash. So these are actually forming one complex polysaccharide or polymer unit. When in the presence of water and in the presence of the digestive enzyme that is required, this will split, and over here there will be split of H plus and OH minus. So from one end H plus will go. So see this one alkyl group, suppose this is R double dash or R double dash, this is going with OH, and one single dash is going with H. So there is splitting up. I'm not showing you all those structures, okay, of glucose, of polymers like starch or units like lactose, maltose. I'm not showing you those units, but if you have read in chemistry or in biochemistry, you are well aware how that reaction is happening. I'm just giving you a gist idea. Now talking about the digestion process, so the disaccharides, they are mainly consumed, that is one is the sucrose, that is the sugar we are consuming, that sugar is the sucrose. Then milk has sugar, that is lactose, and maltose is another type of sugar that is present. So the fruit sugars that we are actually consuming, they are also actually sucrose. Okay, and sometimes they are also fructose. The simpler units can also be present in the fruits, that is fructose can be there, and in the fruits we also can have sucrose. Okay. But remember, all these disaccharides and monosaccharides, the mixture that we are consuming, they will be taken in. Now, if it is starch, a larger or a complex polymer, so that has to be broken, so the time required will be more, the enzymes activity will be more. If you are directly consuming simple sugar, like you are directly consuming glucose, so that glucose will actually not be digested further, it will directly go and that will be absorbed in the intestine. Okay, so that is happening. Now, talking about cellulose, remember cellulose is not for human consumption. Cellulose is not, it means our body is unable to digest cellulose because we don't have the enzyme cellulase. So, what happens over here, coming on to the oral or mouth digestion, here, first, as I've told you, there will be mastication, lubrication of the food, there will be mixing with the saliva. Saliva will be released from the salivary glands and there will be mixing. Now, this saliva, as I've already mentioned, has an enzyme that is tialine. This is also called as salivary alpha amylase. This is also called as salivary alpha amylase. Okay, so this amylase will actually break the starch that has to be approximately 30% at least cooked into maltose and smaller polymers like dextrins. Okay, dextrose, dextrins. So these polymers can be dextrose, dextrins. Okay, so into the smaller compounds. In the stomach, what will happen? So this is what happens in the level of the mouth or in the buccal cavity. Now remember, entire digestion doesn't happen. Certain substances, because we are actually masticating, we are breaking down the food particle for very less duration. We are actually chewing the food for about say 20 seconds, might be 20 to 30 seconds, and after that we are degluting the food, we are swallowing the food, that bolus is coming down into the esophagus. So in the esophagus, as we have already studied, that in the esophagus there is no digestion. Is it? No. There is no secretion of any digestive juice, but the digestion continues. For that time interval, till the food bolus reaches the level of the lower esophageal sphincter into the stomach, till that moment there is continuation of digestion, because there is no such digestive enzymes released from the esophagus, but digestion continues. Remember this. And when it reaches at the level of the stomach, remember, at the pH which is less than 4, any pH which is less than 4, at that level, of course, tialine will get inactivated. Tialine gets inactivated. So in the stomach, there is no activity of any amylase. That is, carbohydrates are not digested in the level of the stomach. Mainly in the stomach, we find proteins slightly broken down by the action of pepsin. Okay, that is released as a proenzyme pepsinogen, and from there pepsin is there. HCl will convert pepsinogen into pepsin, and that pepsin will act on the proteins. Okay, there is no digestion of protein at the level of the buccal cavity. So that will happen, and what will happen, that is the protein will come to the stomach, and there will be the further breaking down of protein into polypeptides. It can be certain oligopeptides also, or even some dipeptides. When I will discuss protein digestion, I will discuss in details. Remember, no amylase is released at the level of the stomach, so there is no digestion of the starch also, because the tialine or alpha amylase gets inactivated at that low pH. The stomach's pH is maintained. When there is food, there is more gastric secretion, HCl is secreted, so the pH is approximately 1 to 2. So at that level, there cannot be any digestion of starch. Whatever the substances, they can be disaccharides, they can be the monosaccharides, they can be the polysaccharides also to some extent, oligosaccharides. So they will be present in the stomach, but they will not be digested. Okay, they only pass through the stomach. This is important. And the thick acidic paste, which consists of the proteins, the fats. Okay, the gastric lipase is also unable to break down larger fat molecules because there is no emulsification. Might be the simpler fatty acids or the small fat chains, they can be broken down by gastric lipase, but other than that, the larger fatty acids, they will not be broken down in the level of the stomach. They require emulsification. At the level of the small intestine, within 15 to 30 minutes, what will happen? There will be churning process, and slowly this chyme will enter the duodenum. Chyme will be released from the level of the sphilotic sphincter. Slowly and slowly, it will be coming into the duodenum. Okay, there will be mixing. At that point of time, the pancreas will also release the pancreatic juice, and the intestine will also release the intestinal juice. So there will be mixing of this entire thing. Pancreatic juice will come, the intestinal juice will come. So there will be this reaction. The maltose that was present, that will be digested by enzyme maltase into glucose and glucose unit, because maltose is formed from 2 glucose units. Maltose is the disaccharide formed from 2 units of glucose. Then we have sucrose. Sucrose, that sugar, mainly we are consuming sucrose, that is digested by sucrase enzyme into glucose and fructose, and lactose from milk, that is digested by lactase into glucose and galactose. So these are basically the simplest sugars. Glucose, galactose, fructose, these are the simplest sugars. So our body tries to actually convert everything into the simplest form so that there is easy absorption. Okay, so this must be clear. Now talking about one particular condition, that is if there is deficiency of this enzyme lactase, that is called as lactose intolerance. In many people who actually might be they are getting older, or from the very birth only, that particular child is not actually, there is no synthesis of lactase, so that child will slowly develop lactose intolerance. Okay, and the unabsorbed lactose will enter the large gut. There will be fermentation by the colonic bacteria, hydrogen gas will be produced, and short chain fatty acids will be there. Lactose, so fermentation will happen, so lactic acid will be produced, so the pH will also fall. So whenever there is lactose intolerance, that person actually suffers a type of, there can be vomiting, there can be cases of diarrhea also. So generally in cases of lactose intolerance, we say that avoid milk and milk related products, okay, because as such you cannot synthesize the lactase enzyme. Now remember, in children we know that they strictly follow the milk diet. Okay, first the mother's milk is given, breastfeeding is done for approximately 6 months after birth, and after that the other milk products are substituted, like cow's milk or goat's milk or buffalo milk, they are substituted. Now remember, in the early phases the child has to curdle that milk, okay, because the chief source of nutrition is that milk, mother's milk. So remember that they have a particular enzyme, okay. The enzyme is actually, in cases of children, they have a particular enzyme that is renin, double N renin. Okay, in adults, in everyone, we have that single N renin that is released from kidneys, juxtaglomerular apparatus. In this case, this is the renin. Okay, so the milk constants, see, lactose, that is the milk sugar. Okay, but in this case, in cases of children, there is a process that is called as curdling of the milk. So that happens, the milk protein is actually involved. So milk protein is there, that is called as casein. Okay, so this casein is converted by renin into calcium paracaseinate. Okay, calcium paracaseinate. So this is the curdling of milk that happens in children. But with age, what happens? This renin actually decreases, and there is also lactase is present. Yes, for digesting the milk sugar, lactase is present in children, but slowly it is mainly present in the But with age, the quantity of lactase actually decreases. So that's why after a certain time we actually develop lactose intolerance. Now over here, see the diagram. In this diagram, we see that over here in the salivary ghrelin, this is getting inactivated at this level. The starch is going and passing directly into the intestine. It is not reacting or doing anything in the stomach. Whatever the lactose, sucrose, if you have directly consumed that, or the maltose or D-xylose. Okay, so all these things are passing undigested in the stomach, they're directly passing into the intestine. And see over here, the pancreatic amylase plays an important role because it breaks down the starch into maltose. Okay, the pancreatic, like there we have seen that there was a salivary amylase, so the starch is broken down by pancreatic amylase. The pancreatic amylase will break down the starch into maltose, dextrose, okay, other units, dextrose, like this, and these will be further digested at the level of the small gut. Okay, and after that the simpler particles, they will be absorbed from this brush bordered epithelium. This is the brush bordered epithelium consisting of villi. So from here the absorption will happen. Now see, talking about the anatomical basis of absorption, first we see that the stomach is a poor absorptive area of the GI tract because it has lacking this villi arrangement. So villi is mainly for increasing the surface area. So surface area is increased, so there will be greater absorption. So that is provided at the level of the small gut, where we have the folds of Kerckring, or they are called as the valvuli conniventis, or they are called as simply villi, microvilli projections. So with this increase the surface area by 1,000 times also at places. The surface area of the small gut, due to the presence of this villi, can reach up to 250 meters square. You can imagine that such a huge surface area is provided at the level of the small gut, so that there is better and better absorption. The continuation of the folds of Kerckring of the valvuli conniventis is until the level of the ileocecal valve, where the small gut ends and there is starting of the first part of the large gut, that is the cecum. Now over here in this diagram, we see the arrangement of the villi. See, this is basically the villi. So here you see that there is blood capillaries present. Into this blood capillary only, directly from the enterocytes, what is happening? The absorption is happening. There are certain receptors, there are certain channels, there are certain other facilitated proteins which are actually helping in the absorption of those nutrients directly into the bloodstream. Whereas there is a central lacteal, basically that is for the lymphatic drainage, where it actually absorption of fat happens. Okay, fats are directly going into the lacteals. There is in fats, in fat metabolism, in fat digestion, we will talk about the formation of chylomicrons, micelles and all, and how fats are absorbed into the central lacteal. Then from there they are passing by the thoracic duct, they are draining into the bloodstream. So that was the structure, the basement membrane. Here see the cross section of the basement membrane beneath the epithelial cells and brush border are shown for this. Per day we say take a several 100 grams of carbohydrate, approximately greater than 100 grams of fat, 50 to 60 grams of amino acid, 50 to 100 grams of ions, because these ions are coming with the water, because water, we are not drinking distilled water, we are drinking the water, filtered water from different like we have the aquaguard and all. So from there we are taking the water, so that has many ions also present. In the food, whatever food we are consuming, they also have these ions present. We are also consuming the nucleic acids along with, if you are consuming chicken, if you are consuming vegetables, so they are all cells. So inside the cells there are presence of nucleus, so there are nucleic acids present, so they will be digested by the nucleases. Okay, they are DNases, the RNases, they are there for digestion. What is this isoosmotic absorption of water? Now see, water in the enterocytes, water is directly absorbed via the process of diffusion. At places there can be osmosis, so from the chyme water is absorbed into the plasma, but at times if there is passing of a hyperosmotic solution through the duodenum, so that means this is a hyperosmotic state or hyperosmolar state. So at that point of time, what will happen? From the plasma, water will be drawn. So water can move either way depending on the gradient, that is the osmotic gradient, that has to be maintained. So water, when it is required, it can move from the plasma to chyme or from the chyme to the plasma. Okay, so this will happen. Now talking about the absorption of ions, this is very important. See, sodium is actively transported through the intestinal membrane. Here you should know that sodium is transported through the intestinal membrane. So 20 to 30 grams of sodium is secreted in the intestinal secretion each day. And for an average, we have said that amount of sodium we are consuming is approximately 5 to 8 grams of sodium. In cases of hypertensive patients, that amount is said to be further reduced. Okay, but on an average, you see, I am writing the data over here. Here it is said that approximately secretion of sodium approximately secretion of sodium that is happening approximately 20 to 30 grams per day into the intestinal lumen, okay, the intestinal secretion, and we are consumption per day is approximately 5 to 8 gram per day. This is in relation with sodium, okay. Now see what will happen. Therefore, to prevent this net loss of sodium in the feces, the intestine needs to absorb the remaining 25 to 35 grams of sodium each day. So this must be done. Wherever significant amount of the intestinal secretions are lost to the exterior, like in cases of extreme diarrhea, when the sodium reserves are lost, so they can actually lead to the lethal levels, means there can be so less amount of sodium that can be lethal. So you know that these ions, sodium, potassium, these ions have to be maintained in the body because they are so important. If you lose these ions somehow through diarrhea, through any other forms, then there will be many sorts of disorders happening. The basic mechanism of sodium absorption from the intestine, that is we will be studying about it, that how sodium is absorbed. Now see from the intestinal lumen, so here what is coming? Water is coming. So water is in normal case, when there is normal, actually there is hypoosmolar chyme that is coming, which has more amount of water. So water will move via the enterocytes into the interstitial fluid, okay. Now see, there is movement of sodium. Now sodium, there is amino acid, sodium amino acid transporter. So sodium will move inside, along with that amino acids will also move inside. So this is basically co-transport. Co-transport is happening, sodium amino acid co-transporter is present. At the same point of time, there is sympone. Sodium is moving along with glucose. So here glucose is also absorbed. So this is called as sodium glucose sympone. This is sympone co-transport. Then what will happen? Sometimes sodium will be exchanged for H+. So there is sodium hydrogen antipode. So here, so when sodium is entering, hydrogen will be released or secreted. So this is basically antipode, sodium hydrogen antipode. Also remember, to maintain the only positive ions are accumulating, so there is requirement of negative ions. So there is exchange. So remember, bicarbonate is always secreted into the intestinal lumen. At the level of the intestine, the pH is somewhere maintained around 7. Okay, so there is requirement of bicarbonate. So there is loss of bicarbonate into the lumen. So in exchange, we have actually chlorine. So chloride is exchanged. So this is basically protein is there. This is anion exchanger. This is chloride bicarbonate anion exchanger. Remember this. Always remember when sodium is entering, so sodium will leave at this level. Sodium always leaves at this level. There is presence of sodium, potassium, ATP is pumped, where 3 sodiums are pumped out actually for exchange of 2 potassium. So this is happening. So here there is ATP is pumped. So this is very common thing that I have been teaching from the renal physiology part, that how this exchange is happening. Always remember, sodium, potassium, ATP pump is present at the level of the basolateral membrane. This is the basolateral membrane, and this side, this is the brush border epithelium, this is the luminal membrane. So this is the transport happening. See, at this level there is basically ATP pumped, so this is active transport. Sodium's concentration is high on the interstitial fluid side, but more amount of sodium is pumped forcibly. There is requirement of the active transport, that is sodium, potassium, ATP pump. Okay. Part of the sodium is reabsorbed along with, at times it is reabsorbed along with the chloride ions also. That is, the negatively charged chloride ions are mainly passively dragged by the positive electrical charges of sodium. So sodium and chloride, they will of course try to actually discharge themselves. So they will actually try to remain closer, but positive will attract the negative, and along with sodium there will be drag of chloride inside. And here also we have this chloride bicarbonate exchanger. Active transport of sodium through this side is basolateral membrane. So here there is the active transport of sodium. Okay, that will happen. That will reduce the sodium concentration inside the cell to a value of approximately 50 milliequivalent per liter. So sodium's concentration inside this cell at a time, what will happen? Over here I am writing that after much movement, active transport of sodium from basolateral membrane. Okay, this will cause what? More and more sodium will be lost to the interstitial fluid. So this will reduce sodium concentration. So reduce sodium concentration in enterocyte. So this is basically the enterocyte, and it will be approximately low to values of 50 milliequivalent per liter. Remember, because of the sodium concentration in the chyme is normally about 142 milliequivalent, okay, and sodium moves down the steep electrochemical gradient from the chyme through the brush bordered epithelium. Sodium is also co-transported with the brush bordered, like there is certain glucose, like we have the glucose transporter, sodium glucose co-transporter. So sodium is co-transported with glucose. So there are presence of the sodium glucose co-transporters, like we have the SGLT1, SGLT2, okay, mainly these two. We have the sodium amino acid co-transporter and sodium hydrogen antipode that I have already discussed. Okay, now see over here, effect of aldosterone. Now we had already discussed the actions of aldosterone. So aldosterone, when the renin-angiotensinogen mechanism is activated, renin-angiotensinogen aldosterone system, so aldosterone has a tendency to increase if there is a fall in blood volume or there is decrease in BP, blood pressure. Then there will be activation of this renin-angiotensinogen aldosterone system, and what will happen? There will be reservation of this water, because there is already fall in BP, there is fall in blood volume. So to maintain that, water is actually antidiuretic effect is lost. So aldosterone, what it will conserve? This will conserve the sodium. So aldosterone, this will conserve sodium, or you can say prevent sodium loss. So when a person becomes dehydrated, large amount of aldosterone are secreted in the cortices of the adrenal glands. Within 1 to 3 hours, this aldosterone actually causes increased activation of this enzyme and transport mechanism for all aspects of sodium absorption by the intestinal epithelium. This increased sodium absorption in turn causes increase in chloride ions also. Water is also moving. So what will happen? Aldosterone, this will also cause water retention. Because what will happen when sodium is absorbed, when the mechanisms are favoring sodium's reabsorption from the intestine, so sodium will always drag water along with it. So there is, because sodium is an osmotically active particle, it will drag water. Sodium also drags chloride ions along with it. So this drag should be maintained. When there is, in cases of if there is dehydration, like in cases of dehydration, what is happening? Water loss, body is losing, the blood volume is actually decreasing, so there is water loss. So at that point of time, aldosterone will be activated, rather aldosterone will be produced from the adrenal gland, and this aldosterone will act on these enzymes, on these certain transporters, and sodium's reabsorption along with water will increase. Absorption of chloride ion also will happen, as you have seen there, chloride bicarbonate exchanger. So chloride exists on exit the cell on the basolateral membrane through chloride channels. So what will happen? Actually, these chlorides, they will further exit over here. So chloride actually seems to exit in this point. Chloride accumulation happens, so chloride exists along with sodium, because sodium is also removed from this basolateral side by the sodium potassium ATPase pump. Okay, so this is happening. In the upper part of the small intestine, the chloride ion absorption is rapid and occurs mainly by diffusion. Okay, that is why following the electronegativity gradient, electrochemical gradient, chloride ions then move along the electrochemical gradient to follow the sodium ions. Chloride is also absorbed by the brush bordered parts of the ileum also, and to certain extent by the large intestine. And also it is exchanged, like here we have seen there was the chloride bicarbonate exchanger. Now talking about the bicarbonate, so bicarbonate ion has to be reabsorbed from the upper small intestine, because large amount of bicarbonate secretion has to be there into the duodenum. Okay, along with pancreatic secretion and bile. So they require the presence of bicarbonate ion. Sodium ions are also moderately amount of H+ they are secreted, and sodium to some extent this is mainly taken inside. There is antipode, as I have already discussed. The H+ in turn combines with bicarbonate, and there is formation of bicarbonic acid. So at this level, if you see this H+ coming out, this bicarbonate, they will combine to form bicarbonic acid, or carbonic acid, sorry, and this carbonic acid will actually dissociate to form CO2 and water. Okay, this will dissociate to form carbon dioxide and water. The water remains in the intestine, but the carbon dioxide is readily absorbed through the blood, and it is subsequently expired through the lungs. So this carbon dioxide diffuses into the membrane, and it is entering into this side, into the interstitium, and from there it goes into the blood. Okay, here in the intestine we actually have basically the mesenteric veins. Into the mesenteric veins it is going, and then it is combining with the splenic vein to form the portal vein. So this blood, it is going back into circulation, and it is expired. So this happens. Okay, secretion of bicarbonate and absorption of chloride at the level of ileum and large intestine I have discussed. Now see over here the paracellular space is cell intestinal lumen. Now see over here, sodium was getting absorbed with glucose. There was that co-transporter that is called as SGLT, sodium glucose co-transporter. So let it be SGLT1. So here what is happening, here sodium is entering, along with this side glucose is also entering. So SGLT1 will transport sodium along with glucose. Then we also have something that is called as SGLT1. Again SGLT2 is also there. So this one will actually facilitate sodium's entry along with galactose's entry. So glucose and galactose, this will be absorbed along with sodium by means of these transporters SGLTs. Okay. Now see fructose, the fruit sugar that was broken down. So this fructose will also be reabsorbed. There is presence of this transporter that is basically GLUT5. Remember, GLUT5 is for fructose. Okay. This side glucose, galactose, fructose, all via the GLUT2. This side we have GLUT2. So all these particular substances, that can be the galactose, that can be glucose, that can be fructose, all these things are passing into the interstitium or into the paracellular space by means of GLUT2. So GLUT2 is present at the basolateral membrane of the enterocytes, and that is facilitating the movement of galactose, glucose, fructose from the enterocyte into the paracellular space. Okay, so this is important, that glucose is transported by sodium co-transporter mechanism, and the facilitation of these GLUTs. One is sodium dependent, sodium glucose transporters, and this side we have the glucose transporter simply, that is GLUT2. And this side for fructose, GLUT5 is present on the luminal aspect, where fructose is actually absorbed into the enterocytes. Now talking about calcium. So calcium's active absorption of calcium, iron, potassium, magnesium and phosphate. Active absorption of calcium, iron, potassium, magnesium and phosphate. Okay, so calcium is actively absorbed into the blood, especially from the duodenum region, and that amount of calcium ion absorption is controlled by the daily need of the calcium for the body. One of the important hormone that plays a role for calcium, remember, that is parathyroid hormone. Now what is the function of this parathyroid hormone? Parathyroid hormone is secreted by the parathyroid gland, and along with that there is vitamin D. Okay, the D, that is the D3, that is the calciferol. Okay, so vitamin D3 and parathyroid hormone, they will activate this process and enhance calcium absorption. So what will this do? This will enhance calcium absorption. So like this, what will it do? It will actually increase calcium's concentration in blood. So that was the function of the parathyroid hormone. Iron absorption is happening. Here I am not going into the details. There is an entire chart that you follow. The presence of metal transporters, the presence of other haptoglobins. We have the ferritin, apoferritin. All these things I am not going into details of iron absorption, but that is very important. Potassium, magnesium, phosphate. There are also other ions that are greatly actively absorbed from the intestinal mucosa. Okay. So with this, remember that iron's absorption If possible, sir, I will teach somewhere that how iron is actually absorbed, but for now just remember that iron is also absorbed from this enterocytes into the circulation, and that is very important. Okay, this actually comes mainly in your biochemistry paper. Absorption of iron is very important. Other ions are also absorbed, and this is the process that I have showed you, that how GLUT5 that is present on the luminal side, GLUT2 is present on the basolateral side of the enterocytes, and how this is facilitating the entry. So with this we end the discussion on absorption of glucose and absorption of digestion and absorption of carbohydrates. Okay, so thank you everyone. Hello everyone, I am Saptarshi Biswas from Murshidabad Medical College, and today we will be discussing about the digestion and absorption of proteins. Now see how these proteins are produced. If you have read about transcription and translation process that occurs in the cells, mainly in both prokaryotic as well as in eukaryotic cells, so you must be aware of the entire process from biochemistry. Now see over there, you know that different amino acids, they join together and form a polypeptide chain. There is formation of a peptide bond. Now this is one particular amino acid. You know that for an amino acid there is on one side you find an alkyl group, one side there is NH2 amine group, and one side we have a carboxylic acid. So that's why they're amino acid. So this entire part is the carboxylic acid. Over here that is similar for another particular amino acid, so that can have this or that can have any other type of alkyl group, like here there can be R or R dash, okay, anything. So over here what actually happens, there is basically a condensation reaction. Okay, in this condensation reaction what happens, there is removal of this water, this amide or this CONH. This is actually called as the amide bond or the peptide bond. So this particular peptide bond is produced by the process of condensation. Okay, so what actually happens over here when these two react, mainly when these react at this level, what happens, this OH and H, from here actually water comes out, this part water is coming out, and they are joining to form this particular bond, that is CONH peptide bond. So this is actually called as the peptide bond, and the groups like this can be R or R dash. So this is by joining them there is formation of the peptide bond. Like this many peptide bonds, like this one peptide, then dipeptides, then like this many units are from polypeptide chains are produced. Okay, and like this proteins are actually formed. Now when we are talking about the digestion of proteins, remember there is a reverse effect, that is hydrolysis. What is hydrolysis? Water will act upon this and this will split this peptide bond. Okay, there will be splitting of this peptide bond, and again there will be formation of this constituent amino acids. Okay, so this should be very clear. Now talking about the digestion of proteins, remember that whatever dietary proteins that we are taking, now see the enzymes that are produced by the mechanism of translation, transcription and translation, they are basically the enzymes required in our body. Most enzymes are made up of proteins. So what will happen, these enzymes will be produced in that manner. The ribosomes are there, the protein factories. So from there the attachment of the ribosomes to the that from DNA mRNA will be produced, and from mRNA proteins will be produced. So that was the entire translational machinery. Now from there the proteins are actually produced, the enzymes are produced, but we are also consuming proteins in some of the other forms, okay, from outside in our diet. So through the diet the proteins are entering into our body and digestion happens. Now automatically in the mouth we are masticating, that is chewing of the food, and after that we deglute the bolus. So that is happening, but remember the bolus is having starch, proteins, fats, minerals, roughage, everything, all the nutrient constituents are included in that bolus, but only the digestion of the carbohydrate starts at the level of the mouth. Okay, there is presence of salivary alpha amylase that will break that some amount of starch for that few moments when it is present inside the buccal cavity. When there is the mastication process continuing, at that point of time certain starches break into maltose, dextrose, okay, and further smaller units, whereas most part of it remain undigested. But remember proteins, fats, they are completely undigested at the level of the buccal cavity. So in mouth proteins are not digested. Coming on to the stomach region, now in the stomach we have different types of cells, like the oxyntic cells or the parietal cells. These cells we all know produce HCl, and I have discussed the machinery of production of HCl. Now that HCl which is produced has a very low pH of 0.8 to 1. Okay, so very low pH. Now what happens, this HCl actually activates this proenzyme, this zymogen, that is pepsinogen. This pepsinogen is also released from the peptic cells. Okay, pepsinogen is produced from the peptic cell, and this pepsinogen is acted upon by this HCl which is produced, and it converts it into pepsin. Now remember pepsin, HCl, all these things, they mix with that particular food bolus which enters into the stomach through that gastroesophageal sphincter. Okay, that is the lower gastroesophageal sphincter. Through that region the food bolus enters. We all know that the action of salivary amylase ends at the level of the stomach, where there is the pH is approximately 1. But what happens, there is dilution. Of course you are drinking water, you are taking in the food particles, so the pH is maintained at about 2 to 3. So estimated pH, this is the range when HCl is mixing with the stomach contents. Okay, and also some amount of HCl3 minus is released by the glands, so that the mucosa of the stomach is protected from the acid. So that also should be there. Now pepsin has two forms, one is the pepsin A and one is the pepsin B. Okay, about which 10 to 20% of the total proteins, their digestion happens, okay, such that they are converted into proteoses, peptones and a few polypeptides at the level of the stomach. So stomach, in the level of the stomach there is digestion of the protein and it is starting over there, but the digestion of the carbohydrates, they are happening at the level of the buccal cavity only, but they are not happening at the level of stomach because there is no action of alpha amylase when the pH is acidic. Okay, but pepsin is acting, pepsin is converting the proteins into forms like proteoses, the peptones, a few polypeptides, there can be the oligopeptides also. But what is the amount? That is only about 10 to 20% of the total proteins taken. Now you know that there is particular something that is called as the collagen. Collagen is very important. This collagen is present. It acts as the animal cement. So it's the constituent of the intercellular connective tissue present in the meats or whatever protein sources we are consuming. They have this high number of collagen. So this collagen is also digested. Okay, so there are enzymes for digesting collagen, like collagenases are present. Now the main digestion that happens mainly at the level of the duodenum and jejunum. Yes, it has started at the level of stomach. It's okay. There are proteases, peptones, a few polypeptides or oligopeptides that are produced, and through the chyme they're entering into the duodenum. Now in the duodenum, we know that the ampulla of Vater, at the level of the major duodenal papilla, there is opening of the long pancreatic duct and also the hepatopancreatic duct. Everything is opening at that level. So there is presence of the pancreatic juice. Now it is for the digestion in the upper parts of the smaller intestine, mainly in the duodenum and in the jejunum. Okay, now let's discuss about the enzymes. What are the enzymes that are produced? So there is presence of these proteolytic pancreatic enzyme. First name and the foremost name is trypsin. These are the activated forms I'm writing. Then we have the chymotrypsin. Then we have the carboxypolypeptidase, and we also have elastase. Now elastine is present, okay, at the level of the intercellular matrix, and also in the extracellular compartment also we have elastase. So to digest elastine, we have the elastase enzyme. Okay, so remember this, the enzyme at the level of the duodenum, that is mainly trypsin, chymotrypsin, carboxypolypeptidase, proelastase, which is converted into elastase. So these are all the activated forms of the enzymes, and they're converting these proteases, peptones and polypeptides from the stomach into further oligopeptides and even to amino acids. Okay, now at the level further below, we have the amino polypeptidase, the dipeptidase. These dipeptidases are acting on the dipeptides. Okay, the amino polypeptidase, they're acting on the oligopeptides, and they're further converting into tripeptide, dipeptide and even to amino acid. One peptide bond is going and it is converting, so from tripeptide to dipeptide, from dipeptide to simple amino acid. Okay, and also remember, in the enterocytes in the cytoplasm, we have multiple peptidases which convert dipeptides and tripeptides into amino acids. So these amino acids are only absorbed. So over here it said whatever I've discussed, that how things are happening, dipeptides, polypeptides and all. Okay, now over here in this diagram, if you focus, you will find that through this region the proteins are entering. Okay, there is acting upon by the intrinsic factor vitamin B12. This is very important step. Okay, vitamin B12 intrinsic factor. Then what is happening, there is carrying of this vitamin B12 intrinsic factor, and they're going, I'm not going into those details how absorption of vitamin B12 is happening at the level of the ileum. Okay, there are different channel proteins also involved in them, and this is very important when you will be studying about megaloblastic anemia. So I'm not going into those details, but for the time being, remember this. Now see these proenzymes which are produced from the pancreatic juice, the procarboxypeptidase, the chymotrypsinogen, the trypsinogen, these are there. Now see how the process is happening. So what actually happens, there is an enteropeptidase released from the intestinal juice. So this is the enteropeptidase. So this enteropeptidase is very important, because when there is an autodigestion of the pancreas is prevented by this enteropeptidase. What is happening, this enteropeptidase is actually produced at the level of the intestine, so it is at the intestinal juice. Okay, but the proenzymes, they are released from the pancreas in the pancreatic juice, so they are not activated at the level of the pancreas, because if they are activated, so pancreas will also be self-eaten or self-destructed. So to prevent that autolysis of the pancreas, what happens, these enzymes are inactivated at this level. What happens, they come to the level of the intestine and they're acted upon by enteropeptidase, which is released from the intestine. This enteropeptidase will act on trypsinogen. Okay, this will act on trypsinogen, and this will convert trypsinogen into trypsin. Okay, now this trypsin, this is very important. When this is formed, what will this trypsin do? Now trypsin will play a role in activating its other brothers, like we have the procarboxypolypeptidase. Okay, so this procarboxypolypeptidase will be converted into carboxypolypeptidase by trypsin. Okay, similarly we have proelastase. So here also trypsin is acting. Okay, and this is converting into elastase. Okay, so this is very important, that procarboxypeptidase, proelastase, these are converted into their respective active forms by the trypsin. Okay, now see what actually happens, there is digestion of the peptide by peptidases in the enterocytes. So at the level of the enterocytes that line the small intestinal villi, so what is there, enterocytes that line the small intestine at the level of the villi, we have, there are branch bordered epithelium which have microvilli present. Now two types of peptidase enzymes. Okay, peptidase enzymes, two types. One is basically the amino polypeptidase, and we also have the dipeptidase. So these are basically produced from the small intestinal enterocytes. These two peptidases, one is the amino polypeptidase and one is the dipeptidase. These will basically further convert the other portions, like the remaining larger polypeptides, into tripeptides, from tripeptide to dipeptide, from dipeptide to amino acid. So body needs amino acid, body cannot work on other things. So in the cytosol of the enterocytes, there are multiple other peptidases that are specific for the remaining type of linkages between the amino acid. Within minutes, and virtually all the last dipeptides and tripeptides, they're digested, okay, to the final stage, and they're formed into the single amino acid that pass, they cross that level. Okay, they cross this level of the brush bordered intestinal epithelium. So ultimately ultimately after every conversion these amino acid they enter. Now this absorption of amino acid, if you know that this was already discussed when I was discussing the absorption of glucose, fructose. Okay, so there are transporters, like we have the sodium amino acid co-transporter. So sodium was entering into the cell along with amino acid. So like this amino acids are actually taken in. So like this protein absorption happens. Luminal membranes of the intestinal epithelial cells in the form of dipeptide, tripeptide, and in amino acid form. So some dipeptides, some tripeptides, they can still be absorbed from the small intestine villi level. The energy of this transport is supplied mainly by the sodium co-transport mechanism in the same way that it happens for the sodium glucose. Okay, that energy is coming from the co-transport mechanism. The most peptides of the amino acid molecules, they will bind to the cell's microvilli membrane with a specific transporter protein that requires sodium binding before transport. After binding, what will happen? Sodium ion will move down its electrochemical gradient to the interior of the cell. So I'm writing over here what is the process that happens. So here the energy required, okay, that energy for most of this transport, that is supplied by sodium co-transport mechanism, like sodium glucose co-transport. Okay, so that absorption is happening at that same, by that same process. Okay, so what actually happens, these peptides or amino acids, they will bind with a particular receptor on the microvilli. Okay, they will bind with the sodium binding, and there will be basically membrane specific transport protein that will require sodium for action. So there are membrane transport protein on microvilli, okay, and there will be attachment. So membrane transport protein on the microvilli, they will require sodium binding, okay. So sodium binding is required at this level. So what will happen? There will be activation, and after binding sodium ions, they will move. Sodium will always move this way. That is, it will move towards the enterocyte. If this is the level, here we have the enterocyte. This is the lumen. Okay. So from the lumen along with sodium, what is happening? There is entry along the electrochemical gradient to the interior of the cell that pulls the amino acid or peptide. This process is actually a form of secondary active transport, because this is a co-transport and here directly energy is not coming. So this process is actually a type of co-transport, and that is basically secondary active transport. Now the difference between primary active and secondary active transport should be clear. Like in primary active transport, like in the case of sodium potassium ATPase form, there is production of energy directly by the hydrolysis of ATP that is moving the sodium against the concentration gradient. Okay, in sodium is high on the interstitial fluid side, okay, but still sodium is forced. Free sodiums are forced from the cell, that is from the enterocyte, into that paracellular fluid, or you can say the interstitial fluid, and to potassium they are brought inside. So this machinery constantly works against the electrochemical gradient by direct hydrolysis of ATP, that is primary active transport. In this case we have the co-transport mechanism. This is a form of secondary active transport, where that energy that is required, that is produced by the movement, and but there is not direct hydrolysis. Okay, few amino acids, they do not require the sodium co-transport, but instead are transported by special membrane transport protein, like in that case like fructose, which was actually requiring GLUT5, transported by facilitated diffusion. So this must be clear. Much is not asked from this part, just you should know the gist of the story, that how this process is happening and transport. I'm not going into the details again, because I've already discussed while discussing about the carbohydrates. So with this we come to the end of this portion, that is digestion of proteins and absorption. Thank you. Myself Satyashri Biswas from Murshidabad Medical College. Today we will be discussing about the digestion and absorption of fats. Now, among the dietary lipids, we know that most part of it is basically the triglycerides or the neutral fats. Triglycerides are basically formed, or the glycerides are mainly produced when the fatty acids react with the glycerol molecule. Okay, so this is a condensation reaction. Here actually water is removed. When 3 fatty acid molecules react with 1 molecule of glycerol, then 1 triglyceride molecule is produced, and this is a condensation reaction with release of water. In the dietary lipids, we also include cholesterol esters, and phospholipids. Okay, and then these will be actually coming into our diet. Throughout that, these are entering into our body. Now, when we are talking the digestion level at the level of the stomach, remember there are 2 lipases. One is the lingual lipase, that is produced from the lingual glands in the mouth, and it requires an optimum pH. Okay, the pH that is approximately 2.5 to 5. That means it is quite suitable for it to actually happen at the level of the stomach, because the pH is actually even it's lesser than 5. So at pH 2.5, at pH 3, this can easily activate itself at the level of the stomach. Similarly, certain cells are there. Okay, these cells, mainly the chief cells. Okay, the chief cells in the stomach, they produce this gastric lipase. So these lipases at that pH, okay, which has optimum pH, it requires, it should be less than, pH should be less than 5.4. So at that pH, what happens actually, there is digestion, but there is very small amount of digestion, only 10%. What are actually digested? These are basically the short chain triglycerides. They can that are present in the milk, the butter or the ghee. They are basically digested. So these mainly digest short chain triglycerides. Short chain triglycerides are digested by the lingual and the gastric lipase, because the larger fat molecules, they require first emulsification, then only they can be further digested. Now, what is this emulsification? Emulsification is basically a prerequisite before digestion starts. Okay, and these the larger fat molecules, as I've already discussed, the larger fat droplets, if this was a larger fat droplet, so this fat droplet has to be converted into smaller forms such that the lipases can act on them, And one of the important emulsifying agent is basically the lecithin. Bile salts are there, sodium and potassium glycolate, taurocolate, but lecithin is also important. Now what is this lecithin? These are also some phospholipids, okay, which includes glycerol, phosphate, choline and fatty acids. So lecithin is very important for emulsification. Bile salts are the detergent activity, okay. Among phospholipid we have this lecithin, okay. Now see the bile salts, what will they do? They are basically lowering the surface tension. So you can write over here, bile salts lowered surface tension, and then they emulsify the fat droplets, okay. Emulsification increases the surface area of the particles for enhanced activity. So when bile salts are lowering surface tension, so what will happen? They will actually enhance the activity of enzyme. So this will cause lower the surface tension, and automatically the smaller fat droplets will be produced, and these will have higher surface area, okay. So this will have higher surface area, and this will help in digestion by enzymes, okay, because the surface area has increased. So what will happen? The activity of the enzymes will be higher. Now see the bile salts, they have a hydrophobic portion, they have a hydrophilic portion. So when we are talking about the hydrophobic portions, so suppose this is a bile salt, this has two portions, okay. This is suppose hydrophobic part, and this side suppose we have the hydrophilic end. Most part of it is hydrophobic. So the hydrophobic part, they are mainly aggregated with the lipids. So suppose if this is the lipid, okay, if this is the lipid, so with this lipid basically the hydrophobic part is attached, and the remaining hydrophilic part is actually attached with that water, okay. So this actually aggravates them to break down into smaller droplets and form the emulsion. The surface area of the lipases will be increased with the help of these lipases, co-lipases and phospholipases. These triglycerides will be broken down into diacyl glycerol, monoacyl glycerol and fatty acids, okay. Like this actually happens, the process actually happens like this. The triglycerides, they are acted upon by lipases, okay, they are acted upon by lipases, or there can be co-lipases also in cases, or there can also be phospholipases, okay. These enzymes will be there, and first they will be broken down from triacyl glycerol to diacyl glycerol, from diacyl glycerol to monoacyl glycerol, and finally to glycerol plus fatty acid. So in this step what is happening? One fatty acid is coming from this step, actually one fatty acid is moving. So like this the fatty acids are removed, okay, and there is formation of glycerol and the remaining fatty acids. So this is the main process with which it happens. Lipolytic enzymes in the intestine, mainly the pancreatic lipase with the co-lipase. Then we also have another class of enzymes that are basically called as cholesterol. We had taken cholesterol esters in our diet, so we have this ester digesting enzymes, esterases, okay. So there are many cholesterol esterases present in the different parts, and they will act. Another important is this phospholipase. So amongst them we have the phospholipase A2, okay. Now talking about the bile, which requires an alkaline pH. So bile actually needs pH of approximately greater than 7.5, okay, greater than 7.5, and the level of the duodenum. The neutralization will also happen because along with bile, bicarbonate is produced. So bicarbonate will neutralize this chyme, acidity of that chyme, okay. And then there will be isomerase enzyme that will shift the ester bond from the position 2 to 1. Now this is a bit of biochemistry. What actually happens, pancreatic enzymes will actually hydrolyze the fatty acids esterified to first and third carbon atoms. So if you see at this diagram, so the carbon atoms, I'm not going into the details of this structure, but just remember that whenever the lipases are acting, they will hydrolyze the fatty acids esterified at the position of first and third carbon atoms of glycerol, forming 2 monoacyl glycerol, and 2 molecules of fatty acids will be produced. Then there will be involvement of another enzyme, that is the isomerase, okay. There will be involvement of an enzyme that will be isomerase, and this will shift the ester bond. What is the function of isomerase? Shifts ester bond from 2 to 1. The bond in the first position will be hydrolyzed by the lipase and forming free glycerol and fatty acids. The major end products of the digestion of triacyl glycerol will be either 2-MAG monoacyl glycerol, or it can be simply glycerol and fatty acid. Cholesterol esters are also hydrolyzed into free cholesterol and fatty acids. So if you talk about cholesterol, these are hydrolyzed by cholesterol esterase into free cholesterol plus fatty acids. And the action of phospholipase A2 also on the that is lysophospholipid and the fatty acids, okay. The phospholipase A2 also produces lysophospholipid and fatty acids. Now the co-lipase, this is the binding of the co-lipase with the triacyl glycerol molecule at the oil-water interface is very necessary for the action of the lipases. The co-lipase is secreted by the pancreas as an inactivated form, okay, and then it is activated by trypsin. Like we had studied over there, that trypsin is an activator of many enzymes like pro-carboxy polypeptidase, pro-elastase. These are all converted into the active forms by trypsin. Even trypsin auto-catalyzes itself. Even trypsin converts trypsinogen into trypsin, okay. So trypsin will similarly activate co-lipase also. So remember this as a point or fact. So if they ask you about the end products of digestion, what can be the end products of digestion? I have told you about lipases, phospholipases, pancreatic lipases. So after this entire digestion, most of the triglycerides will be split up by pancreatic lipases. The end products will be free fatty acids plus 2 monoglycerides or monoacyl glycerol, okay, mainly these. Now see what are the importance of bile salts. They will form something called as mission, okay. There will be formation of mission. Now the products of digestion, monoglyceride, long chain fatty acids, okay. These free fatty acids mainly include the long chain fatty acids. So these are basically along with that there can be some amount of cholesterol also, okay. Some extent of cholesterol can also be present, and also we can have phospholipids, lysophospholipids. We can have phospholipids, lysophospholipids. So these will be there, and these will aggregate to form a mixed mishal. So these mishals are basically spherical surfaces which have a hydrophilic exterior and a hydrophobic interior core. Now what is meant by this part? Let's understand. Over here, if we are getting, this is suppose the fat part, okay, a yellowish fat droplet if I've shown. So this fat droplet automatically is hydrophobic in nature. Now surrounding these fat droplets, we have actually these hydrophilic ends. These are also, see, these taily portions. If I say that this time I've shown you over here in this diagram, suppose this was the hydrophobic portion and this was the hydrophilic portion, okay, but here I'm showing the opposite one. Suppose this long chains, these are basically the long chains. In other words, if I say that this should be a bit different, this portion is the hydrophilic portion, this will attach with water, okay, and this is the hydrophobic portion, because the long chain parts are only the hydrophobic. So if this is the portion, so these are the long chains and this part is the hydrophilic end. So like this. Now water, whenever we are talking about water, so water will act from this side, okay, so there will be bonding with this hydrophilic part, and the hydrophobic part will protect this fat droplet because it is in the interior side, okay. The interior portion is hydrophobic in nature. So this must be clear that what are actually forming this mixed mishal. Mixed mishals, they are formed by cholesterol, monoglycerides, phospholipids, long chain fatty acids, bile salts, everything together combining to form this mixed mishal, okay. Inside the core will be completely hydrophobic. So the bile salts will help this mishal formation and aggregation, and they will be essential for absorption of even fat soluble vitamins also, like vitamin A, D, K, E. So they can also be digested using this thing and aligned at the microvilli surface level of the jejunal mucosa, from where they are passively diffused into the mucosal cells. The enterohepatic circulation of bile salts, I have already discussed that how that salts they were produced and all the re-excretion of the bile salts that are happening. Now re-esterification inside the mucosal cell, what's happening, that once inside the intestinal mucosal cell, the long chain fatty acids are re-esterified to form the triglycerides, okay. Now what is said that when at the level of the intestinal mucosa, inside intestinal mucosa, what is happening, the long chain fatty acids are re-esterified, esterification is happening, they are re-esterified to form the triglycerides, okay. The fatty acids are first activated to fatty acyl coenzyme A by acyl CoA synthase or thiokinase. Now it is a bit of biochemistry. So what actually happens is the fatty acids are activated, these fatty acids, they will be activated, okay, and form fatty acyl CoA by a particular enzyme, that is acyl CoA synthase, okay, not synthase, it is synthetase, or this is also called as thiokinase. And this require, actually whenever we talk about synthetase, at that time, remember that there is basically hydrolysis of ATP, okay. ATP is actually required in this process, 2 energy bonds, so 2 ATP, they're actually producing 2 ADP, and that high energy bond that is basically being utilized, okay. So this must be very clear, and this activated fatty acids then react with the monoacyl glycerol and from the triglyceride. So this will react with monoacyl glycerol and form the triglycerides. So re-esterification happening to form the triglycerides. So this is the process. Free glycerol is absorbed from the intestinal lumen directly into the bloodstream, glycerol, okay, but the fatty acids, they are absorbed into the lymphatics. The free glycerol is not available for de-esterification, and the cells can convert glucose into glycerolphosphate and the 3 molecules of acyl group A that is actually used to synthesize the triacylglycerol. Now see, what are the chylomicrons? This is very important. What are chylomicrons? The triacylglycerol, the cholesterol esters, the phospholipid molecules, along with apoproteins B48 and apolipoprotein A, these are incorporated into the chylomicrons. So what are the constituents of the chylomicrons? Very important. Triacylglycerol, cholesterol esters, we have the phospholipids, okay, then we have apo B48, okay, apoprotein B48, and we also have apo A, that is apolipoprotein A. These are all present into this chylomicron. The chyle, that is basically the milky fluid of the intestinal mucosal cell located in the chylomicrons, they are transported through the lacteals into the thoracic duct. So that is the level of that is absorption is happening. How this absorption is happening? That is basically, if this is the lymph vessel, suppose this is the lymphatic, okay, and this side we have the mucosal cells, and this side is basically the brush border side. So this is the luminal side. Now what is actually happening is that the fatty acids which are actually entering from the luminal side, these fatty acids or the monoacyl glycerol, these are entering from this side, then they are processed, okay. The formation of chylomicrons, they are happening inside. There are presence of the cell organelles, like we have the Golgi apparatus and endoplasmic reticulum. So suppose these are the endoplasmic reticulums and also associated Golgi apparatus. So here the processing will happen at this level. There will be formation of the chylomicrons, okay. The fatty acids, the cholesterol esters, the phospholipids, apoprotein B48, apolipoprotein A, all will be incorporated into the chylomicrons. Suppose these are the chylomicrons that are produced, then there will be exocytosis of these chylomicrons and they will be coming into the lymph, okay. And then via the lymphatic channels, these are basically the lacteals, these are called as lacteals, intestinal level. So they are absorbing this lymph that is milkish, okay, that is a milky fluid that is called as chyle, and that is coming, and then it is passing via the lymphatics into the thoracic duct, and from there it is released into the blood circulation. Is it coming directly? No. So this chylomicron association is very important, because these chylomicrons will have everything along with apoprotein A, apolipoprotein and apo B48, okay. This should be there. So that is about a bit of rough idea about this process. Now talking about over here, if you see the enzymes, so the lingual lipase from the lingual glands, less than 10% of the digestion of triglycerides, Then we have the gastric lipase, also same case. Pancreatic lipase is actually the main, which is actually breaking this triglycerides into monoglycerides and glycerols. And we have something that is enteric lipase, that is the intestinal juice that has the enteric lipase released from the enterocytes lining the villi. And there is no as a significant action because most part of it is done by the pancreatic lipase. So the important one is the pancreatic lipase. Okay. Now over here you see the process of digestion. It's coming. Everything is coming. Over here you see these symbols or notations. Over here we have the cholesterols. We have the cholesterol esterase. We have carotene. We have glycerol. Okay. We have triglyceride, diglyceride, monoglyceride, fatty acids. Everything is coming. Okay. Everything is coming through the diet, passing by from the stomach into the duodenum, and after that they're acted upon by this pancreatic lipase. Okay. This pancreatic lipase will produce this, and after that there will be entire emulsification process that is happening, and after that there will be complete or partial hydrolysis. Micelle formation will happen. These micelles will be transported over here, and inside the enterocytes, in the epithelial enterocyte cells, inside them what will be produced from the Golgi apparatus and all, there will be production of these chylomicrons. These chylomicrons will be then released. After modifications, this will be released into the lymphatics, and they will reach to the level of the thoracic duct. So like this, the chylomicrons that will be produced as a combination of cholesterol, which will have phospholipase, apoB48, apolipoprotein A, all these things will be passed into the, these chylomicrons will be passed into the lacteals, through the lacteals, that is basically the lymphatics of the intestine. Through that they will pass into the main lymph drainage, and it will via the thoracic duct, it will reach the blood circulation. Now over here we will be just discussing in short about a condition that is called as steatorrhea. Okay, this condition is called as steatorrhea. Steatorrhea. Now what is this condition? Actually this is the condition when there is increase in fat excretion, increase fat excretion in stool. What actually happens, what is the cause or etiology? The cause is basically the malabsorption of fat, from small intestine. Okay, due to this increased fat content in the stool, so the appearance of fat, so the appearance will mainly be pale. Okay, this will mainly be pale color because of large quantities of fat present. The fecal volume will increase, volume of feces produced, that will actually increase. Loose texture because of fat, this will have a loose texture, and foul smelling. So this condition is called as steatorrhea. There are various conditions that can cause this, like other causes. Mainly the other causes can also be bile acid deficiency. Okay, then there can be pancreatic enzyme insufficiency, like there can be pancreatic lipase insufficiency. Okay, then there can be also certain diseases. Under diseases there can be certain conditions, that means there can be caused by certain pathogens also, or there can be other things also, like there can be giardiasis, there can be tropical spew, there can be amyloidosis, and celiac disease. Now the treatment will actually depend on the conditions, underlying conditions of the patient. The patients with pancreatic enzyme deficiency, there will be replacement therapy of the enzymes will be given, and other things like dietary restriction of fats will also be there. If someone has problem with the gallbladder, if there is a cholecystectomy already performed, then also in that condition what will happen, there are chances that if the person is consuming more fat, there will be fats present in the stool. That is, fat is excreted along with the stool. So that condition can happen in many patients who have undergone cholecystectomy, and that's why there should be a restriction on the food that they're consuming. Now absorption is probably cleared. Fats cannot be absorbed into the bloodstream because of its hydrophobic nature, so that's why there is micelle formation, and after that there is chylomicron formation, and these chylomicrons are actually entering. The chylomicrons are formed at the level of the enterocytes. The Golgi apparatus, they modify those compounds which are coming after digestion, the fatty acids, even the triglycerides, then the cholesterol esterase, and also esters, and then we have the apolipoprotein A, then we have apoB48. All of these are forming these chylomicrons. There is modification at the level of the Golgi apparatus of the enterocytes, and then they are entering into the lymphatics. Okay, so that was about fat digestion. Nothing much is asked, but mainly remember the process. Most important part of it is this micelle and chylomicron formation, and also remember that you will have to deal this properly. Now if I tell you about something, that is, what is the fate of this chylomicron? The dietary exogenous triglyceride, they will be absorbed and transported into the blood as chylomicrons, as I've already discussed, and these will actually will reach this chylomicron containing, they will actually reach at the level of adipose tissue and liver. Via the lymphatics, they will reach the liver and the adipose tissue. The liver will synthesize endogenous triglycerides also, and they are transported to the liver to extrahepatic tissues. So if it is at the level of the liver, from the liver to the extrahepatic tissue, by means of something that is called as the VLDL, very low density lipoproteins. So these very low density lipoproteins, they will be depositing these extra things into the extrahepatic tissues, and under extrahepatic tissues there can be the adipose tissues also included. Okay, mainly these chylomicrons via the lymphatics are entering into the liver. From the liver to the extrahepatic tissues, they are transported via the VLDL. Okay, during starvation states, what happens? These triglycerides in the adipose tissue, they are hydrolyzed to produce the free fatty acids for energy. When already the glucose content or the glucose reserve of your body is over or exhausted, that point of time, in cases of prolonged starvation. Okay, so at that point of time, what is happening? The adipose tissues, then at this point, they will release these fatty acids. Okay, this will release free fatty acids, and this will be released into the blood. Okay, this will release into the blood. Blood will transport them into complex with albumin, so there will be blood fatty acid complex formed. Then via albumin they will be actually transported, and after that they will reach the cells. Okay, these cells will be there, and then there will be lipid oxidation. Now, how this process is happening, the beta oxidation and all, this will be dealt in biochemistry. Okay, so that was a simple idea about the digestion and absorption of fat. Thank you. Everyone, I'm Saptarshi Biswas from Murshidabad Medical College. Today we will be discussing about the functions of SI and LI, that is the small intestine and large intestine. Now see, the secretions that are released from the small intestine, they were earlier called as succus entericus. Okay, just for your fact, just for your knowledge. Now see, there are certain glands present in the entire GIT. We had discussed about the gastric glands in the stomach. Now in the small intestine, we have certain glands called as the Brunner's gland. Now where are they located? Suppose this is the level of the pyloric sphincter, and the first part of the duodenum, then the second part of the duodenum. So in the second part of the duodenum, we have the opening of the ampulla of Vater, that is, this is the opening of the hepatopancreatic duct. Okay, so at this level, if you take the microscopic structure, then you will find that there are presence of this compound mucous glands. These are called as the Brunner's gland, and these Brunner's gland, when they are mucous glands, they are producing mainly the mucus. Alkaline mucus is produced in response to certain stimuli. One can be the tactile irritating stimuli that can be caused in the GI mucosa. Okay, when it will be produced, when there will be the chyme. The chyme will be released from the pyloric sphincter, so that will come and that can cause irritation. So that irritation will act as a stimuli, and there will be production of alkaline mucus from the Brunner's gland. Or there can be the vagal stimulation. Of course, vagal stimulation seems to increase the Brunner's gland secretion, and certain GI hormones like secretin, they will also cause increase in the level of the secretion. Sympathetic stimulation, remember vagus is basically parasympathetic, so under sympathetic stimulation, if there is any sympathetic stimulation coming, so that will inhibit the secretion from the Brunner's gland. Now see, what are these functions of mucus? Okay, what are the functions of the mucus? Let's write. So mucus is mainly producing over here the bicarbonates. They project the duodenal wall from the the digestion is happening, the highly acidic gastric chyme that is entering into the duodenum, as I've told you, and the ACO3 is released. ACO3 bicarbonate is released, and this bicarbonate will actually cause the neutralization. So this along with liver bile, so bile is also coming. So function of the mucus is mainly bicarbonate release, but along with this bile, all these are totally neutralizing chyme. Okay, these are neutralizing the chyme that was present. Okay, from the food, means the acid was coming from the stomach. Now secretion of the intestinal juices by the crypts of lubricum. Now what are these crypts of lubricum? These are lying between the intestinal villi. Okay, these are basically certain projections into the cells. You can see that these projections have certain cells like the Paneth cell. Then we have the epithelial cells, then we have the mucous goblet cells. Okay, so these cells are basically causing this secretion. Paneth cells, mucous cells, we have the epithelial cells. So the surface of these villi, crypts and villi, they actually contain epithelium. These epithelium consist of cells like the goblet cells and the enterocytes. The goblet cells are basically responsible for mucus production, whereas the enterocytes, they are responsible for production of water and electrolytes, and at the same point of time, they will also reabsorb them from the villi. So that is the general function that we had already discussed. A mechanism of secretion of watery fluid, how watery fluid is coming. If you have read over there, if you have seen that lecture where I taught about that how actively chloride ion or bicarbonate ion is secreted into the intestinal lumen, so you will understand that yes, bicarbonate is produced, chloride is secreted. So these are negative charge particles, so they will ionically drag sodium. Sodium is positively charged particle. So chloride and bicarbonate secreted into the intestinal lumen will be acted upon by sodium. That is, sodium will also be attracted and sodium will also be dragged into the intestinal lumen, and that osmotic movement will also drag water. Okay, the movement of osmotically active particles will also drag water osmotically, and like this the water will also come into the secretion, making the fluid watery. Okay, intestinal fluid will be made watery. Now certain digestive enzymes from the SI secretion, that is the enterocytes are releasing certain secretions, they are digestive juices of the intestinal juice, succus entericus. So that is basically the enzymes include peptidase, then we have the sucrase, maltase, lactase. So there can be peptidase different forms, dipeptidase, tripeptidase, okay, simple peptidase, they are all released from the small intestine, okay, mainly from the level of the duodenum and jejunum. Then we have the intestinal lipase for digestion of lipids, and nucleotides. These are also important because nucleotides also act upon the DNA and the RNA present in the food that we are consuming to break them down. Now along with peptidase, we also have collagenase for digesting collagen. Okay, the collagen which was coming with the proteins, that collagen is digested by collagenase. They're also somewhat extent produced by the cells of the small intestine. Okay, so this was about the small intestine secretion, not in much details, just for your idea. Talking about the secretions of the large intestine, so these are very common secretion, water, mucus, these are always present in each and every secretion. I've told you from the body we will have water presence. Okay, and but remember these ions, I have written over here magnesium, calcium, these actually divalent cations, they are released in the feces. Okay, their excretion happens through the feces. So the composition of secretion from the large intestine, that will be these divalent ions will be present. Now let me discuss about the innervation. Now see the stimulation from the pelvic nerves. So the pelvic nerves from the spinal cord, they will actually stimulate, and these actually when stimulated they will carry the parasympathetic innervation. This parasympathetic innervation will be taken to the distal half or two-thirds of the large intestine, to distal half to two-thirds of large intestine. Okay, and this will cause increase in mucus secretion. So what will this cause? Parasympathetic stimulation. So this will cause mucus production, increased mucus production will be there. Okay, and there will be increased peristaltic activity because the entire GIT has to move, means the contents of the GIT that has to move using this peristalsis. Okay, there is increase in the motility. If there is extreme parasympathetic stimulation, there can be due to any emotional disturbance and all. So there can be so much mucus present in the large intestine that the person has bowel movement of ropey mucus, as often in 30 minutes, where their mucus contains little or no fecal matter at times, but there is only release of mucus. So mucus can be produced in such huge quantity, okay, if there is severe parasympathetic stimulation. Now, functions of the mucus, we all know that is very important for large intestine. If we talk about the functions of mucus, so remember, it has to add that lubricant or the motility so that the stool can actually pass easily from the rectum and the anal canal. So there is basically providing lubrication to feces, lubrication to fecal matter, okay. So this is very important. Along with water, this mucus is important. The mucus adds alkalinity to the large intestine, okay, because of presence of bicarbonate. So bicarbonate will add alkalinity so that there is easier in movement. So this was the importance of mucus. Now see, diarrhea caused by excess secretion of water and electrolyte. Now what is this diarrhea? Diarrhea can be caused by different forms, like there can be a bacterial infection, there can be any sort of parasitic infection or helminthic infection. So these infections are enteritis, that is inflammation of the enterocytes. So mucus secreted large amount with water and electrolyte. Now what is the condition called as diarrhea? There is some difference. So diarrhea, this is basically the Britain spelling, HOEA, okay. So diarrhea basically is mucus plus water, excess amount, okay, excess amount of secretion with stool. But why is this happening? This is reason is basically sort of infection. Now I'm writing this as infection. This can be parasitic infection, this can be bacterial infection, okay. So all these infections can cause increased production of water, mucus and mucus with stool. So this condition is called as diarrhea. Of course there will be additional certain electrolyte losses will be there. That's why when there is severe diarrhea, electrolyte balance in our body is disturbed, and that point of time we have to consume the electrolytes, like basically the ORS, oral rehydration solution, is given. Extreme secretion of chloride ions, sodium ions and water from the large intestine in some types of diarrhea is also noticed, okay. So diarrhea actually happens that certain ions are lost, like the calcium, magnesium, these ions, the chloride, bicarbonate, this will also be lost, okay. In extreme diarrheal secretion initiated by cholera toxin, like if I say cholera is happening, so there is the presence of cholera toxin. So this cholera toxin will actually attack the epithelial cells. This stimulates excess CMP, cyclic adenosine monophosphate. So this will cause the cyclic AMP activation, okay, cyclic adenosine monophosphate, and this opens chloride channel. There is opening of chloride channels. So chloride will flow from inside to the intestinal crypts. So chloride flows from inside into intestinal crypts, okay. Along with, so this what happens, sodium is also moving. So this is sodium drag is happening at this time when excess chloride is lost. So that will also drag, these are osmotically active particles, this will drag water along with it. So this excess fluid is lost to the body, okay. And at times this excessive CV, so this will cause dehydration, this will cause electrolyte loss, okay, and this is quite fatal, remember. Now absorption of large intestine, formation of feces, what is happening? Actually there is absorption of water. What is happening? There is excess absorption of water, so that water is reabsorbed. Now why is water reabsorbed? Because stool is passed as a semi-solid, passed in semi-solid form, okay, and that we call it as your coherent feces, okay. Most of the water and electrolytes of this particular intestinal contents, that is absorption, is happening at the level of the colon, okay, and very little amount of fluid is actually excreted normally. So it is basically less than 100 ml fluid, it is less than 100 ml fluid loss normally, okay. This is a normal scenario. Most of the absorption of the large intestine is mainly occurring in the one half of the colon, giving this portion the name that is the absorbing colon. So remember, the main absorption is happening at the level of the colon. Absorption and secretion of electrolytes, how these are happening, this bicarbonate, chloride, sodium and all these I have already discussed. And it happens that in any case of infection or other substances like bacteria that actually formed as a result of bacterial activity, they can be due to bacterial activity. There will be basically higher quantities of vitamins like vitamin K, B12, thiamine, riboflavin. So they will actually cause, this bacterial activity will actually cause gas production. Various gases will be produced in the large intestine, and this condition is called as flatus in the colon, okay. So this is condition where it mainly contains the gases like carbon dioxide, methane, okay, hydrogen. These gases are produced mainly, and the bacteria from vitamin K is especially important because vitamin K is daily ingested with the food and normally insufficient to maintain adequate blood coagulation if it happens. So vitamin K is also lost, remember. So if it is lost in excess at that point of time, the bacterial degradation will increase in the large intestine, leading to flatus in the colon. Fermentation can also happen. Certain anaerobic bacteria present in the gut, Escherichia coli, okay, certain other bacteria, anaerobic bacteria, if they are present in the large intestine, they will actually cause this gas production. And certain odoriferous, means the odor to the gas that is mainly produced by certain odoriferous products, like there are the indole derivatives, scatol, then we have the mercaptans and hydrogen sulfide, okay. So these actually add to the odor to the gas, that is because bacterial fermentation is happening. So this must be clear. Now talking about certain cases of certain cases of problems like the vomiting. If you know that the vomiting centers are there, the chemoreceptor triggers on CTZ area in the brain medulla for initiation of vomiting by drugs or by motion sickness. So I'm not going into this part because this is dealt in pharmacology. Talking about normally, so vomiting is actually means when the upper gastrointestinal contents, they're actually lost because of reverse peristalsis. What is happening if I say what is basically this vomiting? So vomiting is a process in which the upper GIT contents are expelled out. And why are they expelled out? Upper GIT contents are expelled out due to reverse peristalsis. Now this reverse peristalsis are of course happening due to the nervous. Okay, this reverse peristalsis is happening and there is expulsion of these GI contents, upper GI contents. Okay, so this distension or irritation of the duodenum is actually giving a strong stimulus. So this reverse peristalsis is occurring due to strong irritation or severe distension, or there is severe distension or severe distension to duodenum. So these will actually produce the reverse peristalsis. Okay, other diseases are also there, like this vomiting is a natural reflex also. At times vomiting is also induced. So there will be other forms like, see, vomiting is basically physiological as well as pathological. But here I'm not going into those details. Normally physiological vomiting, this happens at times if there is strong irritation or something. That happens. Even there is CTZ areas, I've told you, CTZ region in the medulla, that is responsible when in cases of motion sickness induced vomiting or drug induced vomiting. And nausea is basically a sensation that is produced before vomiting. So that sensation of vomiting is actually called as nausea. So physiologically these are classified. Then there are many disorders of the GIT. There can be several disorders that are actually dealt in pathology and medicine. I'm not going into those details, but remember there can be ulcerative colitis, there can be Crohn's disease, there can be Hirschsprung's disease. N number of diseases can be there. Adynamic ileus, hiatus hernia, peptic ulcer. Then we have gastroesophageal reflux disorder, irritable bowel syndrome. Many things can be there, which I'm not going to discuss over here. Just you should know the names. If the professor asks you in the Viva table that what are the disorders of the GIT, you can name some of them. Okay. So with this we come to the end of the GIT. Thank you.